You'll see we have our laboratory set up here. John is about to take the mic, and I will pass on to you for an explanation of what all this is about.
Thank you very much, Ineka, and thank you. I shall see how many times I can fall over these lights as well. It's a great pleasure to welcome the BDSRA Foundation, BDSRA Foundation, I'm showing my roots here, to what is now my hometown, even if I'm confused as to where I might be. That's right, I'm easily confused by poker. I play poker once per year. I play it very poorly. It is a matter of pride that I can be the first out. So, anyway, it's a great pleasure to welcome you here. We have brought a whole bunch of stuff from the lab. Some bits and pieces, you'll be able to see those again tomorrow when we have our session. There's a microscope, you can look down them. We have sheep brains, we have mouse bowels, we have all sorts of things. But more importantly, I also have the wonderful people from my lab. If you want to stand up and wave to people. This is my team from WashU, and they all spend all their time working on Batten disease research. They're here to meet you, to listen to you. So please say hi to them and say hello. So let's get started with our science session. So the whole idea of this session is to give you a picture of that journey from what we do in a lab, from the very, very basics, through to the point that we all want to get to, which is having a clinical trial, because all of us in this room would love to have an effective therapy. We'd love to have that magic pill, but how are we going to get there? And the path is not so simple as a straight line from A to B. It's one that takes too many kinks. It takes too long. And some of the reasons why that takes too long is because some of the things we need to know are missing. Pieces of the puzzle that we try to understand simply aren't there. And that in particular is what really goes wrong in these diseases. We might know something about their genetic basis, but what actually really happens inside cells, inside brains, inside the rest of the body, we don't fully know. And that's where we have a whole series of different models. We have some pieces of some of those models here. And we're going to talk about what those models are, why are they useful, what can we do to understand what their disease is like, and what can we do to try and change or prevent that disease. And that's what we call preclinical studies. I'm a preclinical scientist, and that's effectively doing experiments in animals to understand their disease and then try out treatments in them to try and see if they are both effective and safe. And then we want to hand over to a clinical trial. But again, that's testing a therapy as effectively an experiment in people, again with the goal to understand whether they are effective and safe as well. And obviously, there is no guarantee that at the end of that process we will end up with an approved therapy. So we're going to try and walk you through some of this and some of the things that happen along the way. And what are those obstacles that make that path from A to B more windy than it needs to be? Well, part of that is we don't fully understand what happens inside cells, what pathways are the genes we're interested in working in, what do they normally do, never mind what happens when they go wrong. It's also important to recognize these diseases aren't the same as each other. Although they are certain common features, they're not the same disease that is happening at different ages. Each of the different forms has its own specific features and different things, and trying to put them all together is sometimes like comparing apples with oranges, or at least different sorts of apples to one another. So we've been trying to figure out some very basic things like which types of cells are affected. That's why we're interested in cells, which brain regions. We work with a lot of different models that we're going to be going through. Some of those are not ideal, which is why we generate some others, which we'll be talking about. And we have the people here who have generated those models. The larger animal models, both in sheep and in pigs, are here. And we're doing this with a specific goal, which is to try and figure out where can we deliver therapy to be most effective, and what sort of therapy is that. And we'll be going through all of those things. And as we all know, unfortunately, there are other things that happen that slow us down, other considerations, political things, monetary things, things to do with regulatory authorities and some of our partners that sometimes throws a spanner in the works and slows down our progress. And that's difficult for all of us, and we'd really rather wish those things did not happen. So, what are we going to cover today? We're going to start off with Dr. Pearce, who's going to talk about what do Batten proteins normally do and how does that go wrong. He's then going to go on to talk about the different models that we have that we can use to learn more about the disease. Then we'll take over and I will talk a bit about what have we learned from those models and how is that useful. And then Tom Wishart will talk about his work developing biomarkers and what they can tell us. Before we then carry on over, and I will take over again and talk about the preclinical therapies, those experimental therapies that we try out in our models. And that's all intended to lead you through the process up to what a clinical trial is, which Ineka will be taking over and talking about, which will be followed by a Q&A. But knowing that there is not enough time in any Q&A session for everything, we have a session tomorrow, which is an Ask Me Anything session, which is at 1:45, where you can come along and ask any particular question you like, anything to do with the science, anything to do with anything you've heard today, anything about anything really, like maybe you might want to ask, why is it that my English soccer team have got back into the Premier League and Dave Pearce's hasn't? But these are the mysteries that confuse us all. And speaking of which, I will hand over to Dr. Pearce to start the session off.
Nice one, John. Thanks. Okay. There is a break in the midst of all of this as well, so Ineka just reminded me of that. So some of those barriers that John just talked about at the end there, I want to tell you that the fact that these individuals lined up, and you saw they all know each other, they're all working together. The collaboration that we have in the Batten disease field is phenomenal. It really is, and your participation, really helps break down some of those barriers, and you need to recognize that. So where am I looking? Oh, I'm looking down there. Okay, all right. God, getting old is horrible. I can't even read where the buttons are. Okay. Okay, which one is it, John? The big green, oh, of course, the one in the middle. Okay. Well, it's, all right, okay. Basics of Batten's disease. So last year, Kago did this talk, and what we've done is we try to condense things a little bit because obviously it's going to be a very heavy session. So I've stolen some of his slides because I'm known as being very lazy and putting bad slides together anyway. So some of it's Kago's slides, some of it's mine. So it's really, I'm giving him credit, and it's the plus me. Okay, so what exactly is Batten disease? So Batten disease is, you hear this term, lysosomal storage disorder. So the common theme across all of the Batten diseases is that the lysosome has this accumulating material. You hear about things like autofluorescence, GB3, ATP, subunit C. So this is just a simple picture of a cell here right now. So the lysosome is the garbage disposal of the cell. It recycles. That's what that job is. So if you just pay attention to the fact there that that's the organelle or the piece of the cell that's messed up in all Batten diseases. And the brain is exquisitely sensitive to a disruption in that. So in that lysosome there, but also there's a couple of other pieces on there, with other things transporting things there. So pay attention to the fact that in the middle there, you've got scissors chopping things up. So that's an environment where the recycling actually has to occur. But things also have to be delivered to that compartment as well. So what actually happens in Batten disease? So, well, the scissors break. Now, it could be a direct break in the scissors, or it could be something else that's impacting the break in the scissors. But the bottom line is it means that you're unable to recycle things in the cell anymore. So you get accumulation of all of these different things within the lysosome. And then basically it just gunks things up. We put garbage in there because it really is like garbage. It's something that's accumulating. And if you wanted to actually sit down with a bunch of different scientists, we argue constantly about the relevance and what the content of that garbage actually is because it's very complicated. Which gets to some of the things John says. We just don't know some things. There's too many questions we still have. So how does lysosomal function go wrong in the Batten disease? Oops. So I think I showed you. So for the CLN1, CLN2, 10, 13, and we think potentially 5. So CLN1 is something that is involved in prioritizing or tagging things to be broken down in the lysosome. So if that's broken, then you get accumulation of things. CLN 2, 10, and 13 are actually scissors. So those scissors are broken, and that leads to the accumulation. CLN5 is interesting. Some interesting work came out. This is something that potentially it could be regulating in some way some of those other components. So it doesn't have to be that the scissors are broken. It could be something impacting the scissors. So CLN3, 7, and 12, they all sit in that membrane, basically encapsulating the lysosome. And that probably means that they're involved in regulating the contents of the lysosome. So think about it. You hear about things like pH, or hydrostatic pressure, or just the regulation of other molecules. Something about these molecules means something's not getting into the lysosome to allow it to function properly, which means the scissors, they may not be broken, but they're probably not working properly. And then these two here, when there was another thing with transporting things in, there's 24,000 different nuts and bolts in the cells. So obviously it's a highly complicated process. So CLN6 and 8, we believe, are involved in delivering some of those other components that regulate the lysosome. So if you don't have the delivery there, then it's going to be broken and you have the accumulation. I like to think of this as like, if the lysosome is a swimming pool, what's actually happening there is that, and the scissors are broken there, well, someone's basically just been pissing in the swimming pool, and that's why it's broken. So if that's what you should think about with respect to things, is that the environment is just not right. Okay, so we still don't know what all of these proteins actually do. So get back to the basics here. You all know this. We're all made up of 2 copies of every nut and bolt or every gene or protein. So you all have 2 copies of CLN1, CLN2, all the way through the different CLNs. If you have a broken copy and your partner has a broken copy, then you can both have the likelihood is there's a 1 in 4 chance that that broken copy can be inherited by your child. Okay, so what does that actually mean? So we have the DNA, that's the nuts and bolts I talked about, the genes, so CLN1 through CLN14 or whatever. And I just want to say what happens in the cells, and then John will get into some of this in terms of the mechanisms, but certainly some of the therapeutic stuff that we'll be talking about. How can you target a broken piece of DNA? Because broken piece of DNA makes something called RNA, which makes something called a protein, which is, so that's all the nerdy stuff here, and I don't have the time to do a 101, but I'm just kind of trying to simplify it. There's 3 billion letters, which is our code, which forms a template for the program. So the DNA code is the template, and that's the program that goes to RNA that then sends the instructions to make the nuts and bolts, the CLNs that you really want in the cells. So think of it as it's something you read, right? It's basically just 3 billion letters. Actually, and if we actually took a look at that and I read out the code of the human genome, it would take me 9.5 years to read it out. Kind of like one of those lectures that Dr. Cooper gives, right? Okay, bazinga, okay. So the point being the DNA is broken, then the RNA instructions are not there and you get a faulty protein, so which results in the disease. Okay, and you have all this nonsense of letters and proteins, and it's really complicated, so I'm not going to talk about broken sequences there. I'm going to say understanding mutations. Okay, hi, I'm Winnie the Pooh. That's the normal sentence, right? Hi, I'm Winnie the Poop. That's called a change. Sometimes you change the sequence. Okay? Hi, I'm Winnie the Pooh. There's a deletion. Okay? You're missing a piece of the code. So you get the message here is out of all the letters that you actually have, it's very important that that code is right, because guess what? Winnie's just made a bit of a fool of himself. A little bit more lighthearted than obviously your disease. So hi, I'm Winnie the Poohy, and there's an insertion, so there's an extra letter there. So all of those occur in all different rare genetic diseases, and in Batten disease as well. So then you get something that could be totally messed up. I'm willing to poop. So, and that can actually happen as well. Okay, so types of mutation are related to the disease severity. So I've just showed you different ones there. Nonsense one is where we just change something. A missense mutation is a change in the code or whatever. It can be a deletion or an addition. So, because often one of the questions that many of you ask me is the variation in the disease in the children. Because first of all the children are very different. If you take 10 5-year-olds and line them all up, they're all different. Okay? So please never attribute everything that you see in your children to the fact that it's the disease, because it's different. So then when you take the fact that it could be, pooey or poopy or whatever the change might be, those changes can be more or less severe. So that's why your child may be somewhat different to another child. Okay, this is the bit I'm going to whip through right now because John's going to do a really good job of explaining what we've done with the models. So I'm just going to tee this up very quickly. Models are this, something that we use in some of the dishes or in the vivarium or animal facility where we try to understand Batten disease. So we can model that. So then we can have really fruitful studies like the ones that you heard about. We're not going to do anything to any of you guys or your children until we have a really good question that needs to be validated out of the models that I'm about to describe. Okay, simple old yeast, right? Very useful at making bread, but even more useful for making the alcoholic beverages that some of us like to participate in. It's very similar to humans, except for it doesn't have 24,000 nuts and bolts. It has about maybe 4,500. So some of those things could be easy because we can grow them up in 24 hours and study them with respect to what the function might be. So it's used as a model for the following NCLs there. Dictyostelium discoidum, basically it's a slime mold. And this is something, there's a lot of studies you can do there because basically something that slimes around. It's way more complicated than a yeast, so it actually migrates and does things very similar to the types of cells that John studies in the brain. They just don't communicate in the same way. The point being is we can actually manifest lots of studies in yeast and Dictyostelium because we can grow them in a dish in a very short period of time. And there's very little regulations because you can hurt a yeast and a Dictyostelium without any regulations. Worms, so these are C. elegans. If you live in the Midwest like I do, these are you see lots of commercials. They like to eat the roots out of corn, but they're also very good because they actually have some brain cells, very simple brain cells. If you think about how does a worm get to actually eat the corn, it has to be directed. So it has a simple way. So we can do some sensory stuff with that. And why am I saying all this is because John said, and I said, we don't know what some of these proteins do. So we can do way more experiments in these types of models than we can in mice. Flies, so we're getting a little bit more sophisticated now. As we go along, there's more and more of these nuts and bolts. Flies have about 13,000 genes. So again, we can study more and more and what actually happens to the brain of a Drosophila if it doesn't have a functional CLM. Because what we do with these models is we basically recreate Batten disease in those models by taking out that CLN. We have no CLN1 or no CLN2, which is the same as the poopy or the pooey. We've taken out the function of that particular protein. Zebrafish, again, a little bit more sophisticated. Again, if you think about it, obviously we're moving up the chain here in terms of the fact that each of these models has a few more brain cells, if you call it the brain of a fly or a fish. So again, we can study the dysfunction when we knock those things out. Lots of mouse models. This is the bread and butter for us to really understand the brain, and John will talk a lot about some of the pathology there because he's really the world's expert in understanding the brain. But the problem is the brain of a mouse is as big as my thumbnail. Plus, they're really stupid. So we're going to do so much with them. And one of the rate-limiting things you'll find, you hear about the gene therapies. We can fill up the brain of a mouse with gene therapy, but actually translating that and moving up to larger people like children is a challenge. So first of all, we study the cells of the mice to make sure we know which cells we might want to deliver to. So we can mush up the brains, and John's got, there's all sorts of things here you can take a look at, because the brain's made up of billions of cells, and there's multiple cell types, and I doubt we even know what all the cell types are even now, do we? No. So anyway, John's created a sheep model and with Tom as well. So there's the cells that we can study there, because mice, again, they don't have everything that we have in terms of what we want to study. And then, of course, and some of you have participated in these studies, sometimes we'll take skin cells and then try to differentiate them into other types of cell types so we can get a better understanding. Because the bottom line is, the best way to understand a human disease is to be very targeted with your questioning and use some sort of model from a human being. But of course, we can't do a lot of the things we have. So we do use larger animal models to understand the brain in particular. So these are naturally occurring dogs. I think Marty Katz is here somewhere. He's sort of been the champion for many of these particular dogs that have been identified to have defects, and have studied them. These were seminal in the development of Brinura, by the way. So other large animal models, there's naturally occurring sheep, and also we've created models for CLM2 and CLM3, and Tom have created the CLM1 sheep. Now, sheep and pigs, they're not stupid, and they have big brains, so there's lots of ongoing work. The only problem is they're incredibly expensive to upkeep and to study. But bottom line is we're making great progress and having a much better understanding on how to distribute therapeutics in that type of model. And I think the future is very bright with respect to those. Then finally, just because I want to be complete, is there is a naturally occurring macaque monkey that popped up for a couple of different things. Monkey research is incredibly more expensive and highly regulated, but there has been some studies there. I don't think we're quite ready to do some gene therapy studies in these just yet, but that may be something that could actually happen. So I'm going to switch that over to John, who's going to tell you, okay, Dave whipped through all those models. I didn't give you a chance to read them all because we just don't have the time, but John's going to focus and tell you the useful things that we have been able to do with those. Thank you.
Thank you, Dave. So we're now going to move on to, so we have all these models, that's great. What is it we actually use them for, and what can we learn from them? So what is it that we actually want to find out? Well, I think of these as chicken and the egg type questions, really basic things. Where do things happen? How do they happen? When do they happen in Batten disease? And although we're scientific nerds and we like finding stuff out, we're not simply doing this because we want to find stuff out. We're doing this with a real focus that if we can understand the disease, then we can try to do something about it. So we ask very simple questions of those models, which essentially is, how is a model that has Batten disease different to one that doesn't? And we then want to know how those differences change over time. How do they start? How do they develop? How do they get worse as the disease progresses? But very importantly, we have to be able to measure that. And we have to be able to measure that so that we can quantify, because we're scientists and we're nerds and we like numbers, we need to be able to measure accurately how the disease progresses over time. And that's important. Knowing where those things happen tells us where to put the therapy, but knowing how things progress gives us an indication of what would happen if we do nothing about the disease, but we want to put therapy in to stop that. We want to try and treat that disease, but we have to know and be able to measure objectively whether what we have done has actually worked. Is that effective? And just as important as asking if something has been effective is whether it has been safe. We go into doing these experiments with the hope that we will do something positive, that we will prevent disease from developing, or we will be able to treat some of the parts of the disease that have already happened. But it's possible that what we do may cause more problems, and we have to know that out, and we can test that out in a model much easier than we can do. We should never be doing that in people. So starting at the very simple things, we might use cells, and we have some of the bits of plastic here that we grow. We grow cells in dishes, in culture. That's basically putting them in a controlled environment. We can grow them in a dish or in tissue culture, or it's called in vitro, which is fancy language for in glass, but in reality it's actually in plastic, which I don't know what the Greek for plastic is. I'm not sure they had Greek then, plastic in Greek days. But so we grow cells from animals. So many of those animal models that we have, we grow cells from our mice. You can grow cells from the sheep. You can also grow cells from people. Some of the studies that are here are trying to take blood so that we can develop cell lines from people. And those cells can be either primary cells that come direct from the brain of a mouse, or you can preserve them to turn them into a cell line that will keep growing forever. So if we take samples here, we want to take them, we want to then have a line that we can use for experiments for many years. And some of those are reprogramming those cells, the iPSCs that my colleague Patty Dixon mentioned. Those are cells that have the potential to become something else. We can persuade them to become nerve cells, so we can have human nerve cells in a dish. And that's better than having mouse nerve cells in a dish, because they're a big species difference about how things go. And we ask very simple questions again. like we ask of the whole model, how are those cells different to cells that don't have Batten disease? And that can be tricky because some of the tools that we need for finding the Batten proteins are still not very good, despite all the money we've spent over the years trying to make them. But once we figured out what's gone wrong, then we can very easily take drugs and put them on thousands of cells in a dish and ask whether giving that compound, giving that drug, does that make that disease in that cell different? And that's a process of drug discovery. And because you can take gabillions of cells, you can take a lot of different drugs and put them on a lot of different cells and try and find one that does something positive. And that's typically called screening drugs in a drug library. And sometimes that's done with cells, and sometimes it's done with some of those lower organisms, like with zebrafish and with yeast. You can screen different drugs to find one that changes the disease process. And that might give us a candidate that we then want to take forwards and test in a more complicated animal. But at the bottom of all of this, we have to remember that cells are not a whole animal, and a whole animal is not a person. So we have to do these studies with caution and not get over ourselves about the importance of what we've found, and beware that we need to test and retest and see that these things are present across all of our different models to know that they are important features of disease. So that's things we do in plastic. The things we do in living animals, that's what we call in vivo. That's where particularly the mice and the large animal studies become important. And Dave, for example, mentioned how important the dachshunds that Marty Katz developed were for the development of Brineura. And we use a lot of mice and larger animal models. Each of them has their advantages and disadvantages, as we have talked about. But we want to ask simple questions like, where do things happen in the brain? Which cell types are affected? And are all cell types affected the same? And the answer is no, they're not. Some parts of the brain are more vulnerable than others. But that sort of information tells us where we should be targeting our therapy to. And it may well be to multiple places. So, for example, we don't just need to treat the brain, we also need to treat the eye, because we know there are visual problems with the retina. So we need to understand where the disease is actually happening. It's also becoming very clear, and we're doing a lot of work on this, that there is a lot of disease that happens outside the brain, and that is also going to need treating, and we have to understand that if we're going to get a complete treatment for things. But again, as I will say, mice are not people. Sheep are not people, but they are very useful tools for us to try out ideas and to find things, but we have to be cautious not to overinterpret what we find in our animal models. So why do we have so many of these different things? Well, we have lots of different sorts of mice. We have mice that we call knockouts. So a knockout mouse is simply a mouse that does not have the gene. We have disrupted it, we've messed it up, so that it does not make that protein at all. Now, is that really a model of disease? Probably not, but it's a good way to find out how important that gene is. What we do often now, and many of the mice that Dave has developed, the sheep that we have developed, and the pigs that Jill Weimer have developed, are called knock-ins, because they have a particular mistake in the DNA code, and therefore are a direct model of the disease. And that, we think, has advantages over simply taking the gene away, is trying to recreate the mutation that causes the problem in children. So mice are great because they're relatively easy to make. They breed like mice. We understand them. We have lots of tools for investigating them. But as Dave said, their brains are very simple. And often we see that our mice don't go on to get the full range of pathology like we see in the human disease. And that's where having those larger animal models becomes really important. And we have those across multiple species now. And some of those, we had to rely on Mother Nature to come up with a naturally occurring mutation that somebody like Marty had to then spot and identify and develop that into a model. But now we tend to make them, and this is where Tom and his team and what Jill has been doing, is to use gene editing methods, little molecular scissors, that we can introduce mistakes into the DNA to make an accurate disease model. And they are very useful. We have a sheep brain here in a jar. They have a much bigger, more complicated brain. Quite what those sheep use all their brain power for is not entirely clear, but they do have a very complicated brain that is much more like a human brain. They go on to develop disease that is much more like people, and they are very useful tools for us to scale up how to treat a much larger and more complicated brain than is present in a mouse. So when we do those studies, what is it that we want to do? What do we want to find out and why? Well, what we tend to do is we run lots of tests in our mice. So here we have a mouse on a skateboard there. We do tests on them. We look at how they can walk. We measure recordings from their brains to see if they're having seizures. And we try to come up with tests in an animal model that shows something like the human disease, which we like to fool ourselves as being a clinically relevant measure that we can use. So clinically relevant behavioral tests. What I do a lot of as well, we do a lot of behavior, but we also do a lot of pathology, which is looking at cells and how cells are changed by disease and how they die and where this happens and all the different things. And we know that certain bits of the brain are more affected than others. We also like to take brains and mush them up and measure what has changed. That's a lot of the biomarker type work, looking at biochemical changes or looking at molecular changes, changes in gene expression. But what's really important is how you actually do those measurements. You have to do them very rigorously, and how you measure them is really important for understanding, and doing that carefully is essential for making sense of that type of thing. And what we look to do is see how things change over time. And essentially, we're looking to see what happens if we don't treat one of our mice, or if we don't treat one of our sheep, or if we don't treat one of those pigs. What happens in that animal? How do things change? And that's what we would call a disease landmark. And then we're going to come in with one of our therapies, or our experimental therapies, and see if we can prevent those changes or slow those changes down. But also, as I said before, that's the efficacy part. We also have to look to see if anything might get worse. That's the safety part that's really important that we have to look at too. So what do we know about Batten brains? Well, we know that nerve cells die in the brain in Batten disease, and that's a big problem. And we know that some cell types die more than others, and certain populations are very vulnerable, and some of those are ones that do really important jobs. So, for example, there are a whole bunch of inhibitory cells that are meant to damp the brain activity down. We know that they are especially vulnerable across multiple forms of Batten disease in mice and sheep and pigs and people. And we know that that's associated with seizure activity. Some of the work we've done with CLN2 mice recently has shown that there is a direct link between those cells and whether those mice have seizures and then die from those seizures, which we've been trying to treat with gene therapy with some success. So what is it that actually causes those cells to die? Well, to be perfectly honest, we don't know. We know that they do die, but precisely why, we don't fully understand. And we're working on that. That's part of the big grant that Patty talked about that we have, is trying to figure out what actually happens. The first finger of suspicion was pointed at the storage material, that we thought that might be being toxic and killing the cells off. Turns out that's not the case, and it's probably much more complicated. And that big grant that we've got is effectively to look at all the other things that happen inside cells to try and figure out what are the important things that take place inside a cell, which its lysosome doesn't work properly, and figure out what actually does cause those cells to have those problems and go on to die. And, that's why we're still missing that, and that's why we need to do those studies to figure that out, and that's what we will be doing. What we do know is that it's not just the nerve cells in the brain that are the stars of the show. They're supporting cast, a bunch of cells that are meant to look after them and help them. They're called glia. They are very important in the disease process, and they are affected by Batten disease too. We know that those cells become active a long time before the nerve cells die, and they happen mostly in the places where the nerve cells die. And we know now that glia, by growing them in dishes like these, they are affected by disease to the point they can't do what they should do, and they turn rogue, and they actually harm the nerve cells and may injure them and help to damage them. And we use all of these different things, the storage, where the cells are dying, the activation of the glia, as our outcome measures pathologically to see if a therapy have worked. And together with the behavior and the biochemistry, that allows us to test a therapy and see whether we have done something good or whether we have done something that isn't good. And we do that, as I say, with our clinically relevant behavior and focus upon specific brain regions so that we can rapidly and efficiently figure out if something has worked or if something is safe. So we've learned a whole bunch of different things. We know that these diseases are different from one another. They might end up reaching the same endpoint, but how they get there can be very, very different. And we have examples in CLN1 mice, for example. The disease starts in their spinal cord and goes up to their brains, whereas in CLN2 mice, it goes completely the opposite way. And we would think that they might be very similar to each other, but the course in which the same brain regions are affected, but the order in which it happens is completely reversed. And it's important to know that because that is important for understanding whether you've treated something. If you've got two problems that are essentially very different to each other, how do you understand whether you fixed it if you don't really understand the nature of the problem in the first place? So we do think mostly of treating the brain because that's where most of the problems are happening. But we do know that that's not completely effective, even when we get very good at it. And that's because, as we're learning, there are a lot of places outside the brain that are also affected. And obviously that includes the eye, but it also includes many other things in the rest of the body that we now do, looking at things like bowels and muscles that we'll be talking about in a little bit. But I will finish here and hand over to my colleague and friend Tom Wishart from Roslin Institute at the University of Edinburgh, who will tell us about the work he's been doing with his team to develop biomarkers for Batten disease.
Hi. So as John said, I'm from the Roslin Institute, which is the place that made Dolly the sheep, which is one of the reasons why we were in a position to make a sheep model for CLN1 disease. Sorry, can everybody hear me okay? Yeah. Sorry, I feel like a rock star up here. In addition to one of my other roles at the university is I'm co-head of translational biomarker development at the Center for Dementia Prevention, and that is in diseases like Alzheimer's disease as well as childhood neurodegenerative diseases. So what I was going to do is just talk you through a little bit of what we mean by biomarkers, why they're important to know, and what we're doing to try and address those. So as John said earlier on, we have a lot of different animal models that we use to try and understand the processes that are taking place in these diseases. So these give us information on the factors that we can measure to tell us how a disease is progressing. And actually what we need to know then is which one of those things can we use to track not only disease progression, but if a disease is responding appropriately to an effective therapy. So biomarkers traditionally are considered to be biochemical or molecular changes taking place in samples that we can measure. There are things called structural biomarkers as well. So it's basically anything that you can measure on imaging, shape of the brain, size of the brain, or anything which changes at a molecular level in samples. Typically, we like to use things like blood or CSF, as these are considered to be less invasive for the patient. So something like, you might have heard of muscle biopsies. These kind of things are done for diseases like spinal muscular atrophy, but they can be quite painful for the children. So we want to try and avoid those sorts of things if we can. So what is it that we're trying to measure? Well, ideally, in blood, we're looking for things that are turning up in blood that wouldn't ordinarily be there. So these are typically breakdown products of cells that are affected by the disease. So you may have heard of something called neurofilament that's been used before in studies. So these are proteins that are only expressed in the nervous system. And when the cells in the nervous system start to break down, these proteins are released into the cerebrospinal fluid and into the blood, and we can start to detect those things. And the level of those in the blood gives us an indication of the rate of disease progression, or the level of, I suppose, degenerative progression that's happening in the samples. When we measure these things, they can also, in theory, tell us if a treatment is affecting disease progression. So if you see a change in a protein in a patient-specific sample and that patient is giving a therapy, does it start to revert back down towards baseline levels? And these kind of things we need to correlate with other things that we know about disease progression, so disease rating scales or gross imaging of the nervous system using MRI, for example. So these things are really important for us to understand because they're required when we develop clinical trials. So the FDA or the European Medical Agency, for example, have set guidelines on what they want us to be following when we do therapeutic interventions, so that we can tell if we do a trial, is it actually having a beneficial effect? So this dictionary definition is not actually a dictionary definition, it's a definition from the FDA on what a biomarker is, and it's quite complicated, but basically what it means is a biomarker is something that we can objectively and independently measure that tells us about the rate of disease progression and potentially if it's being altered by a therapy. Now, the FDA as well also has a number of different subtypes of biomarker types. And some of these terminologies are quite complex and they overlap a lot. You don't really need to know the specifics of them, but just for awareness, you have several different types of biomarkers. Prognostic tells us what the likely outcomes are of an intervention. Predictive tells us about potential treatment benefits. Surrogate endpoints are essentially a specific measure of the level of effect of a therapy intervention. And monitoring is something that we want to be able to use repeatedly to track with if the disease is progressing in an individual patient, and again, likely if it's responding to therapy. And diagnostic, finally, is basically to say, is there a disease there or is there not? Now, all of these different biomarkers have different levels of utility, and I can give everybody a link to this paperwork if they're interested in reading it. But basically, there's lots of different levels, starting for something in level 1, which basically says if an individual is likely to develop a disease or susceptible to a condition. So these are typically genetic markers. So you might have heard of different forms of a protein called ApoE. If you have ApoE4, you're more likely to develop Alzheimer's disease, for example. It's not guaranteed that you will, but it increases the likelihood. Or another thing would be something called BRCA1 and BRCA2. So if you have that, you're more likely to develop breast cancer. But you're not necessarily going to. It just increases your risk. Right down to biomarkers with higher levels of certainty that we can use to give us firm information on disease progression or status. So for example, something like pressure in the eyeball in glaucoma, or insulin levels in diabetes, that kind of thing. Now, these are not the official 4Bs. These are the 4Bs that we work to at Edinburgh University. So going from the bottom, we have behavioral measures. We have imaging of the nervous system, so CT, MRI, PET imaging. And then for molecular biomarker discovery, we want samples that are either biopsy, so these are quite invasive, but they're specific punches of tissue normally. And we don't really want to go down that route if we can affect it. What we want to use is biofluids. So these are samples that are generally more easily accessible and cause less discomfort to patients to get a hold of. So these are typically things like, well, you might think sweat is something funny to look at, but in the case of Parkinson's disease, I'm not sure if anybody heard about this, but there was a lady in Edinburgh that was documented that she could smell if someone was developing Parkinson's disease because there's something specific in that person's sweat. And they went and did a study and found a marker that you can detect at a molecular level in sweat that will dictate or indicate if you're likely to develop Parkinson's disease. So these kind of things are actually realistic and achievable. They're just not so well known about. But we typically use urine or blood samples as they're much more easy to access. Now, in terms of understanding disease process, that's where the disease models come in, and obviously we're working on CLN1, but it's primarily because we've made CLN1 sheep that we can get samples from. There are other people in the Batten disease community that are working on biomarkers as well, such as David Sleet, Nadia Mitchell from New Zealand, Jill Weimer from here in the States uses pigs for CLN2 and CLN3, and actually we've had a small project that's been funded by the BDSRA, and we've actually been successful in identifying potential biomarkers in blood for CLN1. So I don't know if you can see the blue box on the screen there is CLN1 sheep samples at early and late stages. And on the right-hand side of that, box number 4, is blood samples from sheep that have undergone enzyme replacement therapy treatment that we did with John. So what we can do with that is have a look at the molecular level and try and identify proteins that change with disease progression and hopefully revert back towards a baseline following successful enzyme replacement therapy intervention. And although, as John was saying, all of these models are different and disease progression is different in these different models, we've already had a look at some of the data that Jill's group have generated. She kindly shared that data with us, and we can see quite a few overlaps in the molecular composition of the blood samples between CLN1, CLN2, CLN3, 5 and 6. So this is all very encouraging that we might be able to find something which is useful for tracking these diseases. And alongside that, we're comparing these molecular markers to imaging markers such as measuring the size of different brain regions and the behavioral testing which is going on, as well as visual imaging. And these things, we're now trying to use these collectively to judge whether a therapy is likely to be effective in the animal models. But that's where all of these studies at the start that people were talking about, animal models have their place. But actually what we've got at the moment is a really good indication of if a sheep or a pig is responding to a therapy or gets disease. And what we really want to know is, do those things translate into human patients? I'll leave it there if that's okay.
Thank you very much, Tom. I think you're far more of a rock star than I am. So I shall move forwards to the bit that we're very interested in, which is how is it that we try to develop therapies? And so we're basic scientists or preclinical scientists. So all of the work we're doing is in animal models with the intention of trying to identify something and to return to that theme that might be effective, but that is also safe. That can be a good candidate for being a clinical test. So I'm going to walk you through now what those different things are. So effectively what we're doing in our preclinical work is testing a potential therapy, and that's where the experimental part of experimental therapies comes in, because many of the things we test simply don't work, and it's good that we find out they don't work in an animal model, and then we can concentrate on finding one that does. And the hope is that we will take something that can then go forwards to be tried in people. And to see, there is no guarantee that if it has worked in a mouse or a sheep that it will work in people too. And that's why a clinical trial has to take place. And we effectively have this pipeline that we have. We do proof of principle studies in mice. We then try to take a candidate that works well and then refine the dosing and how to deliver it to a much larger brain. And if then that still holds out and that still works and shows promise and it still appears to be safe, that's what would go forward to the clinical trial. And that's precisely the process that was followed in the development of Brineura, with lots of studies in mice that led to those studies in dogs that led to a successful clinical trial that led to a treatment. And that's what we're trying to reproduce here. But for all of these things, we have to return to, is that therapy effective and is it safe? And to emphasize again, there is no guarantee that anything that starts at the beginning of that pipeline will come out the other end as a shiny treatment. There are a lot of steps along the way, and not anything it can fail at any point along that pipeline. And it's a good question. Should you be going direct from studies in mice to people, or that staging point in between a large animal model we consider to be very important? And these things are not cheap, and we write grants. We get funded sometimes when we're lucky by the NIH, but often we work very closely with industrial partners with the sort of scale of money that's involved for getting clinical trials up and running. So what are these experimental therapies? Well, we're going to walk through each of those individually and say what they are before we talk about them in a little bit more detail. So first is enzyme replacement. We've all heard of enzyme replacement, for example, in the form of Brineura. That's effectively giving back what is missing because of the mistakes in the DNA. And that's a lysosomal enzyme, the scissors that Dave talked about. And that's fine. It's a proven method, but you can only do that if it's an enzyme that's missing. And several of the forms of Batten disease are not mistakes in scissors, like CLN3 disease is not an enzyme, so enzyme replacement is not going to be an option. The alternative approach is gene therapy. So we know that these are genetic diseases, and we know that everyone who's affected is because both copies of their gene have a mistake and they're not functional. So gene therapy effectively is providing a functional copy of that gene using a virus to get inside cells. Viruses are good at getting inside cells and would typically make us sick. In this case, we're using a virus that we've taken out the bits that might cause problems and using it to deliver the genetic message so that it can be made to make what is missing in the first place. We also think a lot of what we call the small molecules. So these are effectively drugs, and these are drugs that, from our understanding of the disease process, will either stimulate something that's not working properly, or perhaps block some of the things that we know that are going on that we don't want to happen and that cause problems. So those may be molecules that act in different ways. It may well be that they were identified by that drug screening process in yeast or in zebrafish, for example. And often we think of taking a drug that has worked in another disease that may be similar, and that's repurposing a drug to try and use that drug and apply it to one of the different forms of Batten disease. Now, there are other methods too. There are methods to edit genes. We use those, like CRISPR. This is what Tom used to make the sheep. This is what Jill uses to make the pig models. But we can try and correct the mistake. Think of this as correcting the genetic typos in that code that Dave talked about. Then there are, very interestingly, antisense oligos. These are changing the message. Dave talked about how genes send out a messenger RNA, which will then be turned into a protein. This is changing that using technology, which is very much at the forefront, very new, to change the message to try and make a functional protein be made. Now, stem cells have been around for a very long time, and the idea is that they, once upon a time, was thought that you could transplant stem cells in to replace brain cells. Maybe that will be possible one day. We don't fully understand how stem cells work, but they do seem to be a reasonably good way as a delivery system for delivering enzyme to where it needs to be. But we tend to use now stem cells more or less of a therapeutic tool and more as a research tool for trying to understand disease biology. Now, all of these different approaches have their technical problems, and those are the sorts of challenges that we have to overcome. We need to get them to the right place, at the right dose, and then again, we have to test, are they effective and are they safe? And I would just like to say a cautionary word. If you look and search on the internet and find there is somebody out there who can cure something, it's probably not true. And beware of that, please. So let's go through these different therapies one by one. We'll start off with the other things. We'll look at the small molecules and then the genetic approaches before we get to enzyme replacement and gene therapy. So a whole range of different drugs are either being tested in animal models or tested preclinically. Some of them have reached clinical trials. Now, it's important to remember that these are not necessarily treating the underlying cause of Batten disease, but nevertheless, they may offer benefit through other different mechanisms. And as I said, that might be by stimulating something that is not working properly, or it might be blocking some of the negative effects of the disease. Or, as I said, they are drugs that are being repurposed that are chosen because they have efficacy in other similar disorders. And a very good example of that is miglustat, that has been used in other lysosomal diseases and is now being tested for CLN3 disease. So I've just put here, and I'm not going to go through all of them, a whole series of different drugs in different categories that might be neuroprotective or anti-inflammatory or stimulate the lysosome or inhibit lipid synthesis or mimic the actions of PPT1. And all of those needed to be tested very carefully in those animal models against those outcome measures very robustly, again, for efficacy and safety. And if you look through the literature from those testing of those compounds, there are a broad range of effects. Some are more effective than others, and only some of those have gone on to be tested in a clinical trial. So how about some other genetic approaches that I mentioned? So I talked about gene editing as a powerful method for altering the DNA, and we hope that we want to alter that back to correct the mistake in the DNA that is causing something not to be made. If we can correct that fundamental mistake, then maybe that can be made. Now, the machinery for doing that, those molecular scissors, has to be delivered to the right place, and that might be through using a virus, but we have to figure out how to get it there. We also don't want those genetic scissors to cut in the wrong place and cause other problems. That's what we would call an off-target effect. And how you control that and how you get that to work, there are new generations. The initial CRISPR, there are newer base editing methods, and there are scientists that are working on NCL mouse models using these type of approaches to refine them and try and get this to work. Now, I mentioned the antisense oligonucleotides, and we have experts here within the room who are doing these approaches. This is changing the message effectively. It is a very specific thing, and it may be very particular to specific mutations, and you have to tailor that exercise, that particular message, the particular antisense oligonucleotides for that particular mutation, and test that out to see if that will work. And that has been clinically approved and shown to be effective in a range of other disorders, so there is hope that this would also be the case for Batten disease too. And there are examples. For example, this is really the potential for personalized medicine to come up with a treatment for a specific mutation, for a specific family. And we know about the studies with Milocen, which was for a CLN7 individual. And there are some very promising studies, and these were done by Michelle Hastings and Jill Weimer, who are both here and will be very happy to answer questions about this, doing studies in CLN3 mice to look at very specific mutations in those mice and the potential for antisense oligonucleotides to be potentially used to treat particular children. So if we now concentrate back on the two main methods that we consider using, which is comparing and contrasting enzyme replacement and gene therapy. So these all depend on simple features of lysosomal biology. So lysosomal enzymes are meant to be inside the lysosome, the cell's waste disposal unit, but they do get released outside the cells and then captured by something on the cell surface and taken back inside. And that's something called cross-correction. Now, what it means, it's just a feature of lysosomal biology, is that if we can supply what is missing, like the recombinant enzyme, like is done in Brineura, there is something waiting to catch it. If we can get it to the right place, there's something waiting to catch it. And not only will it go inside the cells, but it will actually go precisely inside the cells where it's needed, and it should solve the problem. So that's effectively what enzyme replacement is. We have to make that protein that's missing. We have to make it in a dish by tricking bacteria or other cell types to make that protein for us. It then has to be characterized to figure out, what did they make it right? Is it folded up properly? Are all the little modifications that happen to proteins when they're made in the right place? And then it can be tested out. And obviously, we're in a great position now that that does exist for CLN2 through the work that was done through Brian Ura with BioMarin. That's great that that exists. So there are now different options available for CLN2 families. We are working, trying to do something similar for CLN1 disease, and I'll tell you briefly about some studies we've done in both mice and sheep to try and make that happen. Now, one of the issues with enzyme replacement is because that enzyme is used up repeatedly, is you have to keep giving the infusion regularly. And that can potentially cause problems. We have the potentials, difficulties in giving this. This is going to cause an immune response. Might you get an infection or a failure in the port device? And a lot of really good work has been done now since Brian Ura has been a reality to look at those practical issues and how they can be addressed. But it still has to be done, repeated infusions every 2 weeks. Now, gene therapy, this is specifically developed for diseases like this, where you have a mistake in a particular gene. This is using, as I say, a method, a virus, to deliver a functional copy of the gene. And the idea, if we can get that virus that carries that message, that carries that gene that works, into some cells, they can make it for us. It will then be pumped out into the space surrounding those cells, and because of that cross-correction, it will be taken up by other cells. So it means we don't have to get the virus into every cell, we just have to get it in enough to make enough enzyme to make a positive difference. But as we've learned, this method now can also be used for the forms of Batten disease that are not enzyme deficient. So for CLN3, there's been very promising studies done by Jill Weimer, who is here, using gene therapy to treat mice. And if you can get that virus into enough cells, you can have a therapeutic effect within those cells. And this is where there are many people whose careers are making new and improved viruses with different properties to get inside different cell types to be able to treat more effectively in different types of ways. Now, the hope is that in contrast to enzyme replacement, this would be a one-time treatment. We're giving a virus, it will make what is missing for us, and we hope it will do that forever. We don't know that that's actually the case. We don't know that it will keep expressing, and obviously those are the sorts of practical issues we try to address in our models. We also have to be very clear that there are certain toxicity that is associated with some of those viruses. It's also really important that many of those viruses we use are AAVs, or adeno-associated viruses, and at least a third of us in this room have naturally occurring antibodies to those AAVs that make dosing with those viruses complicated. And we have to figure out ways you could mitigate those effects, you could damp them down. We also have to think whether it might be possible to dose somebody again, and how would you stop the immune response that would happen. So there's a lot of issues for this which we still need to work our way through. Now, both of these have been tested for multiple forms of Batten disease. And key question is, where are we going to put these therapies? So, as I said before, we mostly think of treating the brain or the CNS, the brain and the spinal cord, because that's obviously where many problems are taking place. Now, doing that is complicated. We can't simply give these things into the bloodstream because there's a barrier, something called the blood-brain barrier, that stops large molecules from getting into the brain. And that's for good reasons, because we want to protect it from some viruses, but then again, we might want to use a similar virus to treat the disease, and that's a problem. So there are people who are working on trying to make versions, for example, of the PPT1 enzyme that will travel from the bloodstream into the brain, and work like that is going on in a number of places. So generally, while that work is going on, we think of directly treating the brain. But how are we going to do that? That's difficult because obviously the brain is inside a bony box, our skull. How are we going to get there? Well, one of the obvious ways is to drill a hole and to inject that virus or that enzyme you might put directly into the brain tissue. That's something called intraparenchymal. More usual, and what we tend to do more often these days, is to actually put it into the ventricles, the big fluid-filled spaces, and circulate the fluid around and use that to distribute the virus or the enzyme to all the places in the brain that it needs to be. Another way to access the CSF is through a lumbar puncture. That's something that's called intrathecal, and you may see studies where people try to deliver viruses in that route as well. Now, we also have to remember that it's not just the brain. We're also going to have to treat the eye. And we know that if you treat the brain, you don't get as much necessarily of either the virus or the enzyme into the eye. And we need to find ways to effectively treat that. And there are programs that are going on looking specifically at how to deliver either enzyme or to deliver gene therapy to the eye where it can be effective and prevent the changes that we see there. So we spend a lot of our time testing which of the route, or maybe which combination of routes, because it might be we have to use more than one, to get a good therapeutic response in one of our mouse models. And then we try and figure out how to test that in a much bigger brain, like, for example, within our sheep. Not forgetting there is that disease in the rest of the body that we're also going to need to treat too. So I'm going to quickly walk you through some of the studies that we've been doing, looking at testing, for example, enzyme replacement for CLN1 disease, starting first looking at testing gene therapy. We have been doing this for many years with Mark Sands, who is going to be at the meeting tomorrow, and we learned that we need to treat both the brain and the spinal cord. We're also doing studies where we're treating disease outside the brain, treating the bowel and the peripheral nerves, and I'll be mentioning that in a moment. We have been working on enzyme replacement for CLN1, delivering the missing enzyme to our mice. We did a study figuring out which was the best place to put it, and it turned out to be putting it into the ventricles was by far the best. This had positive effects on the disease, and we're now working on ways to make those effects even better than we got. So obviously, we're also interested in developing therapies for CLN2 disease. We know this could be potentially treatable by enzyme replacement or gene therapy, and obviously there is Brineura, which is a great thing. But as I say, that needs to be delivered repeatedly. There are risks of the infections, ports need replacing, and we want to try and find something that will be more effective in the long term. And we've been working with our mouse models, using a mouse that Dave Pearce made and that Jill Weimer kindly shared with us, trying to understand the disease. And we found that our mice actually have seizures that are so severe that they drop dead. And this was all work done by Kago, who gave a talk last year. We found that gene therapy, we can treat those mice, and that prevents those seizures, and those mice live much longer, which is very encouraging. But remembering mice are only so good. We want to treat a bigger brain, and that's because, mice brains are very, very simple. You can see one in a tube here. It's not very big. And that's where we have our large animal models. We see these as a much better testing ground for therapies. And in particular, if we got practical issues of how do we deliver therapy to that big brain, that's where we need different approaches. We can do some very fancy surgical approaches using MRI to guide where we put our therapy and use these as a platform for defining how to dose a bigger brain better. Now, there are naturally occurring models, large animal models. There are the CLN2 dogs that were pivotal for the development of Brineura. There are also naturally occurring CLN5 and CLN6. CLN5 sheep have been essential for the trial for CLN5, treating both the brain and the eye. That was work that was come through from New Zealand. And now, as I say, we can use gene editing to actually specifically make a particular mutation rather than relying on just finding a naturally occurring mutation. And this is where Tom and his team at the Roslin Institute developed those CLN1 sheep that have a very particular mutation. And Jill and her team at Sanford Research, you have developed models, genetically engineered models for both CLN2 and CLN3 disease in mini pigs. And we use those models, for example, using the sheep, to see if we can scale up something that worked well within our mice, to see if we can also treat a bigger brain. And we have done enzyme replacement in our sheep, and by scaling up the dose, we found that we can have positive treatment effects in a bigger, more complicated brain as we can in a mouse, which is encouraging. We can look with MRI imaging, we can look with pathology and see that we are partly able to treat the disease. And we are now working to make this much more effective, and one of the main strategies we're using is a modified form of the enzyme that has better properties to spread in the brain and to get taken up inside cells once we've delivered it there. Now we're also working, and this is in NIH-funded work, looking at gene therapy in the same sheep. One of our colleagues, Beverly Davison, has made us a virus that expresses the sheep PPT1, which we've tested in mice and found to be remarkably effective in those. And we have treated our first sheep last summer. They were treated, and we have more sheep that we will be treating this summer. And we hope to be able to tell you what the effects of that gene therapy will be in the coming years. Now, it's important that we're also looking at disease in other places. This is a disease that doesn't just happen in the brain, it happens elsewhere, and those sites will need to be treated. Now, probably the best known examples of that are things that are happening in CLN3 disease, work on the heart and the altered autonomic system that came out of Denmark. Now, our main interest is in looking at the peripheral nervous system and looking in the bowel. And we think that other parts of the nervous system, if nerve cells in the brain have a problem, components of the nervous system will have problems elsewhere. And we are looking at those. And we're very interested in trying to understand how is that related to disease in the brain, or are there direct effects on other organs like the muscle, and use this as information for putting therapy in the right place. So we work on the peripheral nerves. We look on both sensory and motor nerves. We know that our mice are more or less sensitive to pain at different phases of the disease. We know that there are problems with the nerves innervating muscles, which we thought were actually happening because of changes in the brain, but it might actually be happening because of effects of disease upon muscle itself. And we have been treating those in CLN1 and CLN3 mice and finding some very interesting results that we can improve many of those features of the disease. You've heard me talk before about the bowel. We're very interested, and this is an example of me listening to you. I was told over many years about problems with the bowel in all Batten kids, and we figured out that we should do something about that. And we have found that the enteric nervous system, you have 500 million nerve cells in your bowel. They have problems. Our mice have bowel problems too. They have trouble pooping, and that's because half of the cells in their bowel actually die. That causes those problems. But again, happily, we've been treating that with gene therapy, and we can have a big success. We can greatly improve bowel function in CLN1, 2, and 3 mice. And that's an example of thinking outside the box, thinking outside the brain, that we're going to have to be able to do. So finishing off for my handover to Ineka, obviously this is the preclinical part of a pipeline. We're trying to get to the end, as happened with Brineura, to reach a clinical trial that we then hope might become a therapy. But of course, there is no guarantee of success. Just because it worked in an animal may not mean that it will also work in people. Remember that a clinical trial, while we all hope that it will do something, it is an experiment still. It's an experiment that's been done in people, and we don't know if it will work, just like we don't know if our therapies will work in mice. We hope that something that worked in an animal will then work in a person, but it has to be tested. And it has to be tested rigorously. And that's why it's really important, again, if you take nothing else from this, the importance of testing, does it work, and is it safe, are the basic principles of both preclinical and clinical work. We need to be able to test safety, in particular, safety first, and then has it actually worked. And all of that requires firm foundations. This is why we've spent the time telling you about all of these things we do in mice or do in a lab, because those are the foundations on which a clinical trial are built. So you need to have all of that work done. You also need to understand disease, not just in our animals, but also in people. And that's where we have to have good natural history studies done to give those foundations. And some of the trials that go ahead are not treatment trials, they're actually trials that are done to collect that information. And that finishes me, and I will now hand over to Ineka. 10-minute break, sorry. You probably had enough of listening to me. Have a break, come and play with a brain or something. Thank you.
Okay, if you guys would like to come in and take your seats, we'll get started now. It's time for clinical trial updates. Yeah, okay. So we'll make a couple more announcements throughout the course of this weekend, but we did want to let you all know, if you've downloaded the app, you can now go on and look at the NCL Researchers Challenge. So this is something that we started last year, and we've had a really amazing response this year. It's effectively takes the place of a poster session. So instead of having posters up around on a wall, we've got a more interactive experience online through the app. So if you have a look at, it's called the Researchers Challenge little icon there. And if you click on that, you'll see there's a list of around 20 presentations. We've challenged our researchers across the globe, anyone involved in Batten disease research, to present to us in 3 minutes or less, in 3 slides or less, a summary of the research that they're currently undertaking and what the significance of that research is for patients and families. So you can also vote on that. We'd really love you to get involved with this. You've got 3 weeks to do this. It doesn't all have to be done this weekend. There's a lot more to be occupying yourself with in the room. You don't necessarily have to be on your app for the whole weekend watching these videos, but we would love you to take it home, have a look at some of these research presentations and vote on your favourites. So you can do a star rating as we've got here. And we'll be announcing the winners some weeks from now, and those winners will be featured in our Illuminator newsletter over the coming months. So check those out. Okay, on to the clinical research updates. So this is something that we do each year now. We have quite a number of programs in action right now. So 12 in total programs that I'm going to be updating you on today. I'm always blown away by the fact that this is an ultra rare condition, but the amount of research that's going on and clinical research that's going on right now across the world and some of the big biotechs is really encouraging. We do have some updates. It's a bit of a mixed bag, I guess, this year. We have some not so great news, we have some encouraging news, and just some, run-of-the-mill updates as well as where some of these programs are at. So I want to give you a quick update on those programs. We have representatives from most of the companies that I'm going to be mentioning today from those programs. I'll be giving, so there's many ways we could have cut these updates today, whether we had the companies themselves coming up to give their updates, or if we just, it's a lot of programs and a very limited time. As you can see, the program is very packed for the next few days. So we thought the fairest way to do this is for me to give an update on each of those in a uniformed way, and then to invite those company representatives to come up and answer those questions directly. So if you have any questions from any of the updates that I give during the course of the next 45 minutes, please jot them down in your notebook or write a note on your phone and be ready to ask that question when our Q&A panel come up at the end. And we'll also have the scientists as well that will be answering. So from the presenters this morning, we'll be here to answer some questions as well. So then we'll talk a little bit about natural history studies and registers and the importance of those and some opportunities and some clinical trial resources. And then, as I said, that Q&A. So hopefully we'll have around 40 minutes for a Q&A with our panel today. So John, at the end of the last session, touched on this idea of clinical trial phases and the different studies, the different stages of those clinical trials. We've already worked with the preclinical, so John has covered the preclinical component. That said, there are 2 preclinical programs happening at the moment that have had quite a bit of airtime in social media and through our illuminators and other channels in the last few months. So I'm going to touch briefly on those preclinical studies because of the sheer level of interest there. But effectively, phase 1 to phase 2, 3B, all the way through to phase 4 is my wheelhouse today. Just to give you a little bit of a background, most of you are probably already familiar with this, but the idea of a clinical trial in the first instance is always about safety first, safety, safety. So that is the role of the phase 1 human stage of the clinical trial. So we have a small number of patients, generally in the outside the rare disease world. These are generally healthy adults that are brought in for a phase 1 trial to really look at the pharmacokinetics, pharmacodynamics. So what does the drug do to the body and what does the body do to the drug effectively? And is it safe and well tolerated? Because we're working in such a small population in the rare disease world, we have often combined these phase 1, 2. So in the first instance, we're looking at a smaller population again, but because it's a phase 1, 2, we're looking at a signal of efficacy as well as the safety data. So these can take months to years, and we're looking, today we'll be discussing a couple of programs that are in that phase 1, 2 at the moment. And then we move, if that is seen to be safe, well tolerated, and there's a signal of efficacy there in that phase 1, 2, then those studies will then move on to what we call a phase 2B or a phase 3 or a pivotal. And really that's a larger number of patients, affected patients, whether or not this is like a placebo-controlled or they're comparing to natural history data, whatever that trial design looks like, the phase 2, 3B is really looking at that efficacy, and that's the data that the FDA and other regulatory bodies will be looking at to decide whether or not they want to approve a drug. Has it shown enough benefit to the patients to then move ahead and give that approval? And then we have what's called a phase 4, or the real-world data phase, where the drug is approved, and as you know, Brineura is the only regulatory approved treatment for any of the Batten diseases. It's approved for CLN2, and for some years now, since its approval in 2017 by the FDA, there has been an ongoing collection of this phase 4 real-world data through observational studies, which we'll talk about today, to have a look at what happens to those patients and how is that drug, is it still maintaining its efficacy and safety through the years now that it's approved. So we'll deal with each of those in turn. A quick reminder that this is, as if John and Dave's presentations haven't already, I guess, demonstrated that this is a long, intricate and expensive process to get to clinical trial. We know, and that squiggly line that you showed in the very first slide, Dave, it's not, John, it's not a straight line from lab bench to clinic. It's not a straight line at all. There are lots of whirls and swirls and ins and outs, and the reason for this is, well, multifold, but what we know is that for every 10,000 molecules that are discovered in the lab bench, in that test tube, in that very early stage, only one of those will make it through to a full regulatory approved compound. And the amount of money that takes to get that one compound from test tube into regulatory approval and into human patients with a prescription is about $2 billion per molecule. So this is expensive. It's very risky for a lot of companies, well, for every company, anyone in this field. We are familiar with the term in the industry, familiar with the term the valley of death. And that's really what happens from that stage 2 preclinical, where it's in test tubes and it's in mice and large animal models, moving into humans. There's a huge plummet, I guess, in the number of studies that actually, and molecules that will actually cure or treat, effectively treat. I come from a world of Alzheimer's background years ago, and I can tell you we have cured Alzheimer's in mice many times, but unfortunately we still haven't quite reached the point in humans, and I think now we're down to having said we've cured mouse-Alzheimer's, but that doesn't really mean anything if you're not fixing it in humans. So that's really, I guess, just a quick synopsis. And when we talk about what's happened to some of these programs, hopefully this gives you some context that this is something much broader. It's not just Batten, it's not just CLN1, 2, 3. It's very much, and it's not even just rare disease. It's really right across the field, right across the biotech industry. So keep that in mind for context. Quickly, some definitions. So we're talking today about both clinical trials and IITs, or investigator-initiated trials. The clinical trials themselves are, generally speaking, initiated and sponsored and led by an industry or a company. So this is sort of big investment from biotech companies. Whereas an investigator-initiated trial is very much an independent investigator that initiates the research in response to perhaps identifying the right patient and knowing that there is investigational drug available. And they develop the protocol and serve as the sponsor as the study. So these are generally much smaller studies in a few or sometimes only single patients. And special INDs, so special permissions, are needed from the FDA to do this as well. And generally this is an academic, a clinician, or a small hospital, an institute or a hospital that will sponsor and serve as that responsible party for an IIT. So as you can see, there is a lot going on in this space, and it's a great thing. Like I said, it really, I think it punches well above its weight in terms of being an ultra-rare condition, but having so much activity in this space. It really is an interesting condition, monogenic, so one gene. It lends itself very nicely to a range of different therapies and intervention approaches, as John has very nicely shared this morning. I'm going to go through each one of these in turn. There's a slide or two on each. The ones in purple are actually those two in preclinical that I mentioned, so I'll touch on those briefly. The ones that are crossed out in red are ones that I did give an update on last year, but we have heard nothing since, unfortunately. Whether or not that's an issue, economics or data or what, we don't know. I reached out to every single one of these companies. As I do, we have an ongoing dialogue, some really great dialogue and consistent and, great relationships of integrity and trust. And others, not so much. They might be newer to the field or departing the field, but there will be an update on each of these in turn. I'm just going to go through numerically CLN1, 2, 3, so this is the reason I'm doing this order, and then, not quite alphabetical order for CLN1, but Taysha gene therapies I'll be starting with for CLN1. So Taysha 118 is a gene therapy, AAV9, and as John touched on earlier, AAVs are essentially adeno-associated viral capsids, so they're essentially little viruses where the dangerous material or the disease-causing material is taken out of that virus, and we put in a healthy copy of the gene to basically hijack the cells that it's designed to. The AAV9 comes in quite a different number of flavors, if you like. AAV9 tends to be more of a neuronal specific, so it targets neurons. There are other types of AAVs, like 2, for example, that target other types of tissue. There's a range of them. But in any case, the AAV9 is most commonly used right now in the NCL, in the Batten disease therapies. So this was a program that received IND, so FDA approval in 2019, and in Canada in 2021. Taysha had moved to start a phase 1/2, the CLN1 Connect program for this. But given particularly post-COVID, and this was one of many companies, there was I guess a series of events that followed COVID, and the biotech market was particularly vulnerable to those changes in the economy and the financial crisis, that Taysha was unfortunately forced to pause their enrolment of that program, and it has been on hold since early 2022. We have, however, had a great relationship with Taysha. They've kept us in the loop. We've had regular meetings throughout all of this time and remained in good communication. And we know that they are actively still exploring opportunities to enable the continuation of this program at the clinical trial level. That said, we know that earlier this year, the Batten disease patient advocate groups around the world came together, had worked with Taysha, and had worked with the investigator here that's relevant to this announcement to bring together this global statement to say that there was some promising progress in the Taysha 118 program. And this was announced by us on February 15 in collaboration with Taysha, and shortly after Rush University Medical Center in Chicago also released this really great press release. This is online on their site. What did it say? So an individual patient IIT was granted to Liz Berry-Kravis, who unfortunately is not here with us today, but one of her clinical trial coordinators is with us, and she'll be able to answer some questions on this as well in the Q&A. A 7-year-old child with CLN1 disease was dosed with Taysha 118 in October 2023, and they were treated at Rush Medical Center. And it's been, I guess, something that the family community have been waiting for. I think this was very welcome news, but very, I guess, tempered news, because this is only in one child at the moment. So we are some months in now to this child being treated. We know he's doing reasonably well. I know Stella will be able to share a little bit more on that when she comes up. But essentially now, they are looking at further evaluation of the efficacy and safety data from this first patient, and we hope that there may be opportunities to expand this cohort to enroll some additional patients. On CLN1 as well, you may remember from last year, we talked about the treatment of CLN1 through a blood-brain barrier penetrating vehicle. So it was an enzyme replacement therapy that actually was given IV, so intravenously, and because of the technology that was employed, that it's able to cross the blood-brain barrier. And this also was an IIT. This was an investigator-initiated trial in Germany in collaboration with JCR Company. And the German patient, he was 3.5 years old. This was on compassionate use. And they found, I always say we were, a global community. There were no serious adverse events, and there was some efficacy, some sort of, again, an N of 1, you can't have statistical significance. It's not powered to show statistically significance in terms of benefit to the child, but we did see some efficacy and improvement in quality of life, and some of those psychosocial measures were improved. Unfortunately, that poor family, that poor child passed away from unrelated causes, which was pneumonia, about a year after he'd started that. The company took the decision to no longer pursue that particular molecule, that particular treatment program, the AGT194. But what they have, I only spoke to them on the phone earlier this week, they're very much still engaged in their CLN1 program. They are now exploring alternative modalities. That's sort of, I guess, the extent of the detail that I was given, leveraging what they call the J-brain cargo. So this is a blood-brain barrier penetrant technology. Whether that cargo they're moving across is the TPP1 enzyme or perhaps a gene therapy, I'm not clear on what that is, but the signals are, I guess, encouraging in that they hosted a small family meeting to coincide with World Symposium earlier this year in San Diego. And they really wanted to discuss the plans for a CLN1 natural history study, which is really important data to be collecting for clinical trials. They participated then shortly after in a global roundtable meeting that the BDSRA Foundation organised with a great number of external stakeholders that are all interested in the CLN1 space. And really we felt it was our duty to bring all of the interested stakeholders together, knowing we're having conversations with some companies, some institutes, some academic research institutes that are doing, small natural history studies. We really wanted to bring everybody to the table to have that conversation, and we thought we were best placed to do that. So we'll talk a couple of more external stakeholders that joined us with that meeting as well. But JCR is certainly in the picture, and they have offered co-funding for any natural history studies that do move forward in this space. Again, preclinical, in this case, collaboration pharmaceuticals are NIH funded for a lot of their research and development at the moment, and they're currently undertaking IND enabling toxicology studies, meaning they want to see that this ERT, this enzyme replacement, is not harmful in the animal models that they're testing, and look at that efficacy in their animal models and bring that to the FDA to say, could we please run a clinical trial? We think we've got something here. They also held a small family meeting in February. There was a lot going on in February, and this was very coincidental, I imagine, which is why we said, right, we really need to do this roundtable because there's all of this happening in silos. So they have now moved ahead with collecting family details through their own CLN1 natural history, registry. We'll talk a bit about the differences between a registry and a natural history study shortly. But the Global Roundtable meeting was another opportunity for them to bring their information to the table and talk about opportunities to work together. So this was the roundtable, and we were really proud of this. Some of us down in Australia had to wake up at about 2:30 a.m. to participate in this 2-hour meeting. Coffee is always my best friend at that time of day. But it was a 2-hour meeting. We had lots of good, healthy conversation and discussion points and ways that we could practically and pragmatically move forward. The thing I hate most in this world is just blowing hot air around a table and everybody just talking, wouldn't this be nice? This is a great idea, and then never actually getting back together again. That's just not how we operate with BDSRA. We like to get stuff done, and that's definitely, we definitely found some points of synergy here to get these parties working together, and hopefully for the betterment of you guys, most importantly. So you are at the centre of everything we do. CLN2, BioMarin are here in the building. As you know, there's quite a few of you in the room. Always a great signal and a great. I always talk about optics in terms of companies being present, and they have been with us through the journey for some years now, 10 at least, and are here today, so they will be able to answer questions on these. But as a quick here we go. So John, you already touched on this. The enzyme replacement therapy is an ICV, so intracerebroventricular, meaning it's going into the fluid pockets in the middle of our brains and replacing the enzyme through that, and the uptake of the enzyme happens through the tissue over the course of that infusion cycle. So the phase 1/2 was completed some time ago, but now the open label extension, meaning those children that were on the trial are now, so the 23 patients from 3 to 16 years are continuing to be followed through this. The 5-year data is now published. Angela Schultz from the Hamburg UKE team published this earlier this year, and effectively what they were able to show through this paper is that over time it is safe and well tolerated, that continues, and that we see this stabilization. So while there is some decline, it is the stabilization remains for that. We're up to now 6 years of data since the beginning of the first child being treated. So we're seeing the longevity of the slowing down of the motor and language scores with children treated with Brineura. There's also a 10-year, so this is the phase 4, again, we talked about real-world data, a 10-year prospective observational study, again, really just, and this remains in the recruitment phase, so new children coming to diagnosis and initiating therapy will be enrolled particular sites around the country, internationally, and they will be collecting data on these children for years to come. And this is all part of the due diligence that every company, every responsible company should be doing in the real world. Then we have the ocular ERT. So this is something that's received quite a bit of media and, well, social media attention. Certainly within our community, there was a lot of discussion around the fact that for a long time we knew that the ERT wasn't making it down the optic nerve and into the eye, and the children were still going blind on the ICV infusions. And so this study was started a couple of years ago at Nationwide Children's Hospital. This is a sponsored study from BioMarin by invitation only. It's now fully enrolled with 5 patients aged 2 to 6, and those children have one eye randomised. So one eye is treated, the other one acts as the control to see, well, what happens when it's not treated. And this has been a 2-year study at Nationwide. Dr. Emily Delos Reyes is in the room and will be available to answer questions on that as well. The safety data was presented at World in February 2024. At the moment, the efficacy data, again, because it's a small study, the efficacy data remains to be seen, but what we do know is that it's safe. There have been no serious adverse events in terms of infections and intraocular pressure and so on, sort of the usual safety measures that are investigated. At the same time, similar timing, over in the UK at the Great Ormond Street Hospital in London, Professor Robert Henderson and Paul Gisson commenced an IIT, so an investigator-initiated study that was effectively something driven by the BDFA. Liz Brown-Nutt is in the room somewhere, the president of the BDFA. Talk about what a group of families and advocates can do when they get together and fundraise and drive a really important agenda. And they were able to start this study with what is effectively termed leftover drug. This is not a BioMarin-sponsored study, but this had 8 patients randomised. They were all the right eye treated. The silver lining with degeneration in CLN2, as with most of the NCLs, is that the eyes seem to degenerate in a very what's the word I'm looking for? Bilateral. So both eyes will degenerate. It doesn't seem to be one degenerates quicker than the other. So you can quite comfortably and reliably use the other eye as a control to see what happens when one's treated and one's not. So this was actually published at the very end of last year, this particular study. And what they found was effectively that it was seemed to be safe and well tolerated, no issues in terms of infection, sort of serious adverse events or increases in intraocular pressure and so on. But these children were a little bit older, and it didn't seem to have a huge effect in slowing down that photoreceptor loss or retinal thinning. But again, because they were older patients, and really the takeout from this was not just the safety data, but that it would be preferential that the children would be treated earlier in the course of the disease to see if there was a better sign of efficacy in those younger children. So Lexio, this is really, I guess, a very brief, and I know I am just hammering through these. I'm hoping that once your subtype comes up, you can be really engaged, and then when your subtype passes, you can kind of, yep. But there is no other way to do this unless you wanted me up here for 6 hours, and nobody really wants that, so I probably can't understand half of the words I say anyway with my sloppy accent. It's not quite the Queen's British. But the... so in 2020, this was in...
Not British.
I'm not even allowed to say, okay, English. Australian English or British English, maybe that's what I need to delineate, versus US. So in 2020, the phase 1/2 was actually in clinic. So we had a phase 1/2. There were 8 children treated with this in 2020. This was an intraparenchymal administration. So when John showed you the slide of the dots on the brain, effectively they would drill holes in the skull and inject it straight into the meat of the brain, the flesh of the brain. It was safe and well tolerated, but compared to what was happening in the ERT space at that time with Brineura, the rescue of the motor and language scores and the decline was not as significant as what was being seen in the ERT. So they chose to pivot, go back to preclinical and look at alternate routes of administration and different dosing. So they chose to try in non-human primates going in via that little intracisterna magna, which is another pocket of CSF fluid at the back of the brain. They did find it had a better uptake, better distribution, and there was an increased expression of the TPP1 at clinically relevant concentrations. However, once that preclinical data went back to the FDA, they said that they really needed to do further preclinical studies before they could move back into the clinic. And I think at that time, all factors considered, biotech market, economy, competitors in the space, they made the decision earlier this year to no longer pursue this program, and unfortunately they're seeking partners to take on the program, but we've not heard anything since, and I think they've quietly moved off into other programs. So, but that's by the by, because in the meantime we know there's been a lot of interest, and this was a different vector as well in Lexio. The patented technology with the AAV9 with REGENXBIO has seen 2 programs in recent years. We know that the ocular, so they have 2 programs, ocular and CNS, so one into the eye, one into the brain. This is subretinal, so it's going right into the layer of the cells within the eye. A phase 1/2 was initiated. This was 4 patients were dosed in Great Ormond Street Hospital in London. And that first patient was dosed last August, so shortly after the conference here, and we sort of knew that this was coming and this news was on the boil. We weren't allowed to share anything at that point, but that first patient was dosed in August. And was being followed, and since then another 3 patients have been dosed under the protocols under this particular program. Unfortunately, we did suffer a blow as a community. It was hard. There were a lot of tears from our team and from the REGENXBIO team too. But the CLN2 programs were halted due to strategic pipeline prioritization and corporate restructuring. Unfortunately, euphemisms that we've heard far too many times in this industry. However, the data collection for this is still ongoing. We have remained in touch with REGENX, and there'll be more on that in a second. I just want to cover the other program with REGENXBIO. So this was an individual patient IIT again, so hopefully you're familiar with that concept now. They're sponsored by an individual clinician or research institute, a clinical research institute, so in this case in Brazil. The 6-month data was presented in August at one of the Society for Inborn Errors of Metabolism meetings, and they found that this particular therapy in one child this child was 5 when he was dosed. And they'd found from this child that it was well tolerated and safe. There were no serious adverse events. The sustained levels of TPP1 expression were encouraging. So over time, it's now 1 year past his treatment. And as far as we know, from these data, that the TPP1 gene expression is maintained. There are increased intervals between the ERT, so this family felt that they no longer needed to have every 2 weeks an ERT, so infusions for Brineura. And he had clinically meaningful improvements, so 86% reduction in his seizures, and leading to withdrawal of 2 of his anti-epileptic meds, and improvement in fine motor and expressive language. So again, N of 1 efficacy is very hard to say, with any confidence, you need a much larger study for statistical significance, but good signals to begin with. November 10, the news came, or November 8, we sent out a joint statement on November 10 to say, so that we wish to assure you, Regenex's letter to the community was they wanted to ensure us that they would committed to working to find potential partners to ensure these programs advance. Amy and I met with Ken Mills, and it was really, I guess, the best of a really bad situation in that they wanted to keep those communication lines open with us and believe that the gene therapy has the potential to impact the course of Batten disease. So this is not a clinical data issue, this is not a safety issue, this is an economics issue. They've been committed to stewarding this program. We have remained in touch. The fact that Vivian Fernandez is here from in an unofficial capacity, but Vivian has been on this program. Vivian, do you want to give us a wave? Can you stand up and say hello? But she is here. We've had a really great relationship with this company, and they will not step away until this program has been stewarded forward and shepherded well. And we know that is certainly happening. I can say hand on heart that is, when they said they were committed to moving this forward, that wasn't just hot air. It was very much the case. We've had an April 9, we had an Ask an Expert with Paolo Falabello, who is the clinical lead on this program, and he took the punches and took the hard questions and knew they were coming, but wanted to give, the reassurance that they were still in looking to, swinging to move that license on to the next company to continue that program. And that data continues to be presented. May and February this year, we've seen the 381, the ocular program, presented at World and at the ARVO conference. Again, with the TPP1 expression, we know that it's increased and sustained in the treated eye, and there was preservation of photoreceptors in the treated eye, again, small n of 4 in this particular study, but the retinal degeneration seemed to be slowed down compared to the untreated eye. So that's, again, the fact that you guys are here speaks volumes, and I really don't want us to lose hope and become despondent in times like this, because it is hard, but we're all in this together. And so thank you for being here. Preclinically, Lattice Bio. So we're up to CLN2. We're going to roll. We've still got 10, 15 minutes left, but we're going to roll through the others quickly. This is classic Ineka right here. The Lattice Bio, so this did receive a lot of attention. It's actually a novel proprietary AAV, so we're still talking viral vector, but it's a different flavor again. It's not a 9, it's their own secret AAV capsid. We're not privy to that at the moment, but they're currently going through the motions for pre-IND and to have that regulatory approval to start clinical trial. They worked with us to run that survey, you remember, back in March, understanding the patient journey. And I just want to highlight the importance of your participation in these kinds of surveys. When we put these out, they're not just to take your time away from your family and children and to give you menial tasks to do. These are very pragmatic. We use this data. We put this data together with the Lattice Bio team, and they actually used that, and they said it was really particularly beneficial and instrumental in supporting their application for this regulatory approval to move forward, because it expresses the needs of the community that we're actually trying to treat. And this stuff is taken seriously more and more so by the regulatory bodies. So we hope that you would trust us when we put those calls out, that we wanting to steward your input to your benefit as far as possible. CLN3, Amicus, very briefly, we know Amicus CLN3 has been floating around for a while. We've had updates on that in recent years on where this phase 1/2 study has been. Unfortunately, in February 6, Amicus made the announcement that they were going to be returning all of the rights, so the legal rights and licensing back to Nationwide Children's Hospital, along with the CLN6 and CLN8 pre-clinical for CLN8. Emily Delastrez will be available to answer questions as far as she can in the Q&A. CLN3, so this is really the only active study right now in this subtype. So Batten 1 is, the active ingredient is miglustat. Miglustat is a substrate reduction therapy, so looking at an ingredient that goes into the junk that builds up inside the lysosome. It's effectively taking out one of those ingredients in the hope that the rest of the junk will stop building up. It's, you can't bake a cake without a key ingredient. Well, this is a key ingredient for that junk. It's an oral tablet that has been approved for use in other lysosomal storage conditions. So it's approved for Gaucher's disease in the US, and in a number of other markets, but not the US, it's approved for Niemann-Pick type C, both pediatrically and in adults. We have been, so when I say we, disclaimer, I also work in a similar role for the Beyond Batten Disease Foundation. So we all work very closely together, our organisations, but we had spent quite some years' time and resources building the preclinical data to show that this does seem to work in CLN3 mice and improve the phenotype, improve the clinical presentation. So we had the go-ahead to move forward with the phase 1/2, which has actually just finished. So the last 18 months, this data has been collected in adults. So again, this is a safety-first study, but because there were 6 patients in here, we were able to collect some efficacy data, as in we're looking for signals of efficacy, not statistical significance, but certainly looking at those signals. We know that it's safe and well tolerated across the time frame. There can be some GI side effects, but these can be well managed with dietary changes, and timing of dosing is important. But we know at 12 and 18 months that there's been a reduction in very key neurodegeneration markers, neurofilament light chain, and we've seen a reduction in that glycosphingolipid, which is part of that fatty junk building up inside the cells. Clinically and functionally, we've seen that there's been a decrease in that progression of motor decline as measured by what we call the UBDRS. It's a rating scale to have a look at all the different domains that a patient might have deficits in over the course of the disease. And whereas the untreated, historically data from natural history studies showed that there was a decline, in a 6-point decline in untreated over the same time frame at the same age, and for 18 months, there was only a 1.8 decline, 1.8 score decline in the treated patients. So that data collection's now complete, and the manuscript's in progress. Those 6 patients that were treated will still continue to access miglustat through a compassionate use program for the foreseeable future as we move into phase 3. So now we're looking at the paediatric study, which was due to commence earlier this year, but unfortunately, another announcement that there were some unanticipated delays with this. Theranexis had had some issues with an interested investor, and essentially unrelated to the program. It was something to do more with the other investor on the other side of the fence, having to pull out it unexpectedly, unbeknownst to all of us that this was coming. So unfortunately, we have had to pivot and look at how we might get this to approval in the fastest time possible. It's really important to convey to the families that this isn't a massive biotech company that's reprioritizing pipelines and shuffling things around. This is very much, for both Theranexis and Beyond Batten, this remains a lead program and the highest priority. And we're here because this program is here, and it's not going to be one of those cases that this program is shelved and moved and reprioritized elsewhere. So you heard that, it's recorded. We're in it, we've got skin in the game, and we're moving forward with this phase 3. At the moment, we're looking at ways to redesign that study that we might actually get it to market or to approval in the shortest time frame possible by collecting off-label use. We know there are families that are using miglustat off-label, and we're trying to use that to our advantage. But certainly there'll be further updates coming down the line. We have a dinner tonight for CLN3 families, and stay tuned because we will communicate with you whenever and however we can as soon as news comes to light. CLN5 has been a great news story. This is a gene therapy that is now moved to enrolling a third cohort in their phase 1/2. So 6 patients are now, three patients are now fully enrolled in the low dose and the mid dose range, and they are now recruiting for the very high dose range. Or high dose for another 3 patients. And this is happening in 2 sites, both in University of Rochester and in Great Ormond Street in London. So they plan to share that data in the second half of this year, and we know that they're planning on moving forward to a pivotal phase 3, so this will be a larger study with more children at whatever dose they deem to have been the most safe and efficacious, and that will be sometime in the next 6 months. They're meeting with the FDA to discuss some of this safety and efficacy data and figure a plan on how to move this forward. They've also had some great, outside of the Batten program, they've had some great success with their RET program as well. So it's been, and they've had a recent merger acquisition. So it's really good, I guess, in terms of optics and signals from the company. This is a healthy company moving forward and certainly engaging with us regularly as well. So skin in the game with this program. They also have a natural history study, which is now fully enrolled, but just to let you know that this is something else that they've invested in. CLN6 disease, again, this is something we've been hearing lots of in recent years. This is a program that's really been driven from the ground up by a family foundation, the Charlotte and Gwyneth Gray Foundation, and they'd been working with Nationwide and then on with Amicus to move this program forward. 13 patients were actually treated in this phase 1/2 that were at 1 year or older, and those children continue to be followed. Unfortunately, Amicus was part of the package that went back to Nationwide, but that continues on. The children are still being followed, and the Gray Foundation, I spoke with Kristen just the other day. Unfortunately, as many of you know, Charlotte passed away on the 4th of July. I spoke with Kristen some days later, three days later. She called me, and whereas I thought I was going to be getting a very bereaved mum on the line, which of course she was, wow, is that woman just, it's like a fire was lit under her, and she is damn dead determined to keep moving this forward, and this will be Charlotte's legacy, and for Gwen as well. So I think just watch this space, Dr. Emily. But there's definitely, it's an amazing story, and we're really grateful for the family foundations, like the many that work together to do this. CLN7, so this is the investigator-initiated trial from UT Southwestern that's been going for a couple of years. The safety data was first presented just a few months ago, well, back at the end of 2023 in Hamburg, and really the safety data shows that there's no side effects or serious adverse events. At the moment, so the nerve conduction was stable and improved following treatment, but beyond that, the efficacy data still remains to be seen. But in the meantime, they've actually partnered with a company called Elpida. So UT Southwestern have partnered with Elpida, who are operating in a voluntary capacity. They're really very much a social enterprise company, volunteered to help bring together external stakeholders and help move forward with with the natural history studies, because this is really important, and we're about to talk about why natural history studies are important. But they're helping UT Southwestern to move this forward to a phase 2, and we're hoping that more families, CLN7 families, will engage to participate in that natural history study, just to get on their radar that if they do move forward with that phase 2, that there's patients that they're aware of around the world, because this is, of course, an ultra rare form of Batten disease. So natural history studies in the last couple of minutes. Why are they important? You've heard about them this morning. You've heard our research team stand up here and say they're recruiting. Please come fill out a form, come fill out a survey, come see us. We want to measure you, and all of the different domains. Well, why is that? Just some of the reasons. They help us understand the nature of progression. Remember, we're rare. We're ultra rare, and there's not a lot. We are as good as the data and the knowledge we collect along the way with our families. It helps us forecast prognosis, so better management, better preparation for what's next along the disease journey. And it's essential for drug development. Like, this does not happen without you guys. We cannot get to the point of a good, robust clinical trial design that doesn't need a placebo-controlled arm where your child is receiving, you know, a sugar pill as opposed to the actual treatment of choice. And it also helps us evaluate how effective that data is, how effective that intervention is. Here's a really nice example. So the Brineura clinical trial, so the solipinase alpha clinical trial, was conducted in 23 children. They collected this data over 2 years for this particular trial. And as you can see here in the blue line, the motor language score across the course of that, on average, for those 23 children, was this blue line. So it didn't seem to change too much. But that doesn't mean anything if you're not comparing it to something. What does the untreated child look like? So this is where the historical controls came in. And once you put that red line in, suddenly it makes a difference. Suddenly you can see a really clear, distinct difference in this particular therapy that it's having compared to the children historically that didn't have that. And if they didn't have this historical control, they would have unfortunately had to do this placebo control, which means they would have had to recruit another 23 kids with a progressive terminal illness to sit in a trial for 2 years and receive a sham injection, or, you know, something that's not the active drug. And there is talk of that in the FDA. There is talking of sham injections to sort of act as a placebo. It's horrifying, but this is why the natural history data is really important. So see the study team. Trust us when we say we've got, you know, when we say we've got opportunities for surveys, registries, and natural history studies, please get in touch with us. The family register is in the app. You can go on to, it only takes a few minutes. The difference between a register is really collecting very top line information. Some will go a little bit more detailed, but you don't actually have to go into a clinic and have your child measured and, you know, hand over all of the blood samples and the EEGs and all of the different tests for the different domains. This can be done generally through surveys and through your own recollection of your child's disease and progress. You can find out about clinical trials on our website. We have an interactive page, and we are always updating this. Every month in the Illuminator, we're updating this. And there's some great clinical trial resources that we're going to pop up on that page as well. So the American Society for Gene and Cell Therapies is excellent, just for general background. If you're sitting around twiddling your thumbs with nothing else to do, as I know you all are as Batten parents, go and have a look, because they really do have sarcasm. It's Australian, very Australian. And the clinicaltrials.gov also offers some really great resources as well. And of course, I'm here 24/7. If you need any, no matter how big or small, I'm here to answer your questions, as is all of our team. So let's move on to the Q&A. And I'm going to just do a roll call of who's here to bring them out the front.
Okay, while you're doing that, I was going to have the first question, just so we can have a clarification from the Rush team in terms of the CLN1 patient that was treated in that N of 1 is actually 7 years old, which is relatively old. So I just want to make sure we understand why that child was eligible.
Okay, so before then, can I have Emily McGuinness from Taysha to come up? Stella Bockman from Rush? Yeah, so Dave, they're coming up.
Yeah, so you can answer that question.
Stella would be the best person.
It's a IIT. So Stella's going to answer the questions specific to the IIT, and Emily is here to answer questions on the broader program. Guillermo Cerati from BioMarin, please. Dr. Emily Delos Reyes from Nationwide, Craig Benson from Beyond Batten. John, come join us, please. Tom, if you've got questions for the scientists, or maybe the scientists can answer some of the trial questions too, who knows? And then Dave, you're here as well, but you can rove whatever you like.
I'm the runner.
I will hand over to you guys. We have like 40 minutes, so take your time, please. And you're welcome to stick around
so
once
Stella,
lunch
if you
is
could answer
dismissed.
that first question, I think that's an important question for the audience to know.
Yeah, so the first patient was 7. It is thought that he had juvenile onset of CLN1. So.
Well, I think part of this is that we didn't really talk about eligibility criteria, so why was that 7-year-old eligible for this trial? Well, for the N of 1, I should say.
Yeah, so he was identified as being a good fit for the trial, mostly because his progression was still. Sorry. Yeah, so it was thought that his progression was still initial, and it was
So it's
less.
mildly affected. The reason
Yeah.
being is usually most CLM with children at age 7 are very, very further progressed. So I just want to make sure we understand that, what the eligibility, when is it more treatable with respect to that? That's all. That's the clarification I was looking for. Okay, if I'm not being blinded, questions from the audience?
So I have read that focused ultrasound has been successfully used to temporarily open the blood-brain barrier. And I was wondering if anyone is looking at that in relation to CLN diseases, Batten disease, and whether or not that would provide some new opportunities to develop or deliver therapies.
So that is a possibility. Obviously, I talked about the blood-brain barrier as being an obstacle. It's there for a good reason to prevent viruses that might do harm into the brain. So any way that can bypass that barrier, and we sometimes do that by simply ignoring it and putting things directly in the brain. We heard about the study that was using carrier technology, so linking the enzyme to something else so that it would get across into the brain. And we heard about the work from JCR Pharmaceuticals, that IIT, an N=1 study, and they're now using different technology. Now, the approach you mentioned is a different way to do that, which is to actually disrupt the blood-brain barrier, and people have done that with drugs. They've used drugs to do that, and those drugs have potential problems. So the idea of using ultrasound to do it is another potentially safer way to do it. To my knowledge, no one has yet tried that. We would be interested in talking to the people who do that so that we could try to do it. Now, it may be there are practical limitations on how to do that that would make it difficult or more technically challenging to do in a mouse, but perhaps a larger animal model might be well suited to doing that. So we have a large animal model of CLN1 disease. There is a large animal model of CLN2 disease, perhaps might be best suited for trying out those approaches. So I'm sure that would be a useful discussion to have with the people who can do that, so that we might try and see if that would work. But we would need to partner with those people. Most of science is a team sport. Life is a team sport. Science is a team sport. That's the so we would have to get together and work with that. Dave talked about how we like to collaborate. We'd love to have a chat with those people and see how to do that.
Anybody else on the panel want to add to that? Okay, now I've learned I've got to keep away from the speakers, so.
Sorry,
Okay, Tom.
Sorry, I was just going to add, so our human clinical neuroimager had talked about that before, but I think in his opinion, the term disrupt could be substituted for the word damage, and that's always risky for something that's supposed to be there for a reason. So potentially, but there are risks.
Okay, Amy has a question.
So I'm curious, I think I first heard about this in World, but with the enzyme replacement, and I'm not smart enough to know about the others, but when we see an immunoresponse, why are immunosuppressants not being used? And is that something that everyone's looking to do?
So in our CLN2 trial, which actually we're part of the Lancet trial, it's not just UKE. America is also involved. So yeah, 4 centers. So the Lancet article is nationwide in London and Hamburg and the Italian. So I want to give proper, just in case they hear that, oh, we were not mentioned. So anyway, so why do we not use immune suppressants? So when we started this study about 2014, so whenever we give infusions, we also pre-medicate. And that was also part is adverse events, right? So, for example, the most common is IVIG. We standardly give Tylenol or Benadryl to prevent side effects. And like the big guns now are called MABs, right? So when you have MABs, that's like magnificent when they have side effects. So part of what we did was we very carefully administered this intraventricular enzymes in the ICU. These kids had to stay in the ICU for 24 to 48 hours during the time that we were studying this for about 3 years. So we've watched these children if they had side effects. Majority of these children did not have side effects, and we kind of reacted. So we saw now, I've heard of kids having some rashes. I think I see this, and I've heard about this in much younger kids. The minute in the children with the CLN2, a fair amount of them were already moderately involved, but at the same, so that means they were older. But we did not see a whole lot of reactions to the enzyme. So for example, some of them probably have what we call stop mutations, and they did not, surprisingly enough, they did not have this reaction. And when they measured antibodies, we did find antibodies, but we did not see a drop. For example, if you have antibodies, that means you're reacting to the drug, right? And you're not going to get a good response. We did not see that. So we had the same, you saw Ineka's graph, and they all still had the same response. And the same thing with the eye. We're not seeing, so it's not so immune mediated, I guess, this drug, because we're not also seeing any inflammation. So for example, when in the intravitreal trial, we have people in our DSMB where they're all eye people. Because what they can have is they can have vitritis, or I'm not probably saying it correctly, but they didn't have any of that. So far in our 5 children, we're not seeing that, it's pretty amazing that these children that we have, although again, there's only 5 children, that we're not seeing enzyme reaction. So we did watch these children. I talked to a lot of physicians all over the world. When there is a reaction, then we react. We say, okay, give them steroids. We usually use Solumedrol or whatever it is that is in their location. And then sometimes the initial dose may not help. We even double that dose. We try our best to kind of suppress that reaction. And probably I've heard that in my mind, I don't know, maybe 2 or 3 at a time. I think there are some families here that the one child I know has a rash sometimes when she has her, I'm kind of winking at the mom, when she has her infusions.
John?
So speaking from a very basic science perspective, obviously there is a potential if this individual, whether it's an animal or a person, has never made this protein before, when you give it to them, the chance is there for an immune response. Now, it turns out that different proteins are, to use a fancy word, which I don't like to do, immunogenic to different extents. So different enzymes provoke different types of immune responses, and those immune responses also differ whether they're within the brain or whether they're within the eye or anywhere else in the body. So obviously when we do our ERT studies in animals, we look out to see are antibodies raised against these enzymes and are they causing a problem. And our mice similarly have Benadryl. They don't have full-on immunosuppressant because we try to keep things as simple as possible to not have too many variables. Our experiments are expensive enough to do, enzyme versus no enzyme, that adding immunosuppression on top of it becomes even more complicated. So we don't see, using PPT1, any great immune response against that. That doesn't mean that we don't look out for it, and particularly when we jump a species boundary and we go to sheep, then we have to look again, because sheep have a different immune system to mice, to people.
Go ahead, Mark.
So I would say, to complement what Emily and what was said here, is that in our clinical trials, and this was published, we monitored the antibodies throughout the clinical trials, and there were no correlation between the positivity or negativity of antibodies or the level of antibodies, low, medium, high, with any safety or efficacy. So, and that's why it's not monitored in the real world, and each patient, whenever they have an adverse event, needs to be individually evaluated. In other ERTs, some cases, it's not very common, they have had severe reactions, anaphylactic reactions, And some of them had to go through what is called a desensitization protocol to make them tolerant to the ERT. And some of them needed those immune modulations that you mentioned, but those are very rare cases. And that's where you team up with specialties like immunology to help you figure out what's the nature of the reaction and what are the measures that need to be taken in place.
So as a follow-up, we have kids that have been on it 6. So as a follow-up, we, again, not a scientist, but if we have children who've been on it for 6 and 7 years now. So the longer that you're on it, does that give them more opportunity to build up antibodies against it?
Not necessary, not necessary.
Yeah, the immune system's highly variable. When I spoke, I talked about the variation in children just with respect to the manifestation. Every biological factor of human beings can be very variable. To John's point on the immunosuppression in the animals, just so you know, it's actually easier for us to knock out the immune system genetically to do some of these studies rather than keep giving them drugs. So that's the basis of some of the things we do. Questions? Okay. Now I've learned to stay in the middle of the room. We can no more.
Hi, thank you for putting this together. This is so lovely, and I'm so grateful you are all here with our families. I want to back up and just ask a quick kind of general topic. We're in 2024, so we're in the age of artificial intelligence, and that certainly affects all parts of our world when we search things on our phone to when we try to call and get a prescription for our kids. We can't talk to a real person. So I was curious, looking in these rare spaces, how does partnering like the human brain with artificial intelligent technologies, what are some ways from the preclinical lab work all the way through trial, collecting data, through all of that, what are some ways that the AI technologies can be helpful or can be utilized?
I gave the microphone to Craig, and he gave me a deer in the headlights. I mean,
yeah. Thanks a lot, Dave. I appreciate
but,
that.
I mean, he's innovative and is always on top of things. So I'm sorry.
Yeah, I guess
I can give a quick answer to that.
Okay, all right, here. Then
So
John.
we evaluated some initiatives to do AI, for example, to identify patients through medical records, teaching the systems how to look for red flags in the medical records, and trying to help identify patients who could be compatible, for example, in this case with CLN2 or MPS, look at x-rays and scan all those medical records and try to look for signals and help to see where potential patients with a similar phenotypes as CLN2 are living, to help with the identification of patients. That's a way that AI can help. That's one of the ways that I could think about.
Craig?
Yeah, I was just going to say on the spectrum, and this is a more general commentary and specifically related more to the capital markets for biotech and biopharma, not to mention the rest of the technology world. So we're generally speaking in very early stages here of the AI game. And it can be a great tool, will be a great tool. In fact, I would venture to say of all the business plans and fundraising packages I've seen in the past 8 to 9 months, every single one of them has some AI component to it. Now, so I think the real challenge with AI is finding very specific utility for it and then applying it within that specific scope. And so I think particularly in this space, how to apply that is still under development for the most part, because it's just this, how do you boil the ocean? I mean, it's that kind of size issue, but also that kind of sized opportunity. So I mean, it's clearly going to have an impact. I just don't know that we can begin to even know right now how all that might happen. So,
John?
Yeah, so we are also using AI, and this is central to that large NIH grant that my colleague Dr. Dixon talked about earlier. We're trying to look across multiple lysosomal diseases at multiple different parameters that change and compare them all at once to each other, which is beyond my ability. It's beyond most of our abilities, but we have experts at WashU who work on those machine-based learning, AI-type approaches to be able to compare things across multiple different modalities in multiple different ways to make sense of it. And for example, we have systems, we have colleagues where we can teach them to recognize specific features of disease in cells in a dish that we can use to be characteristic to say this is a disease cell, this is how sick it is, or has it responded to therapy. So we're using those approaches and those technologies already. We're also using machine-based learning approaches. I now have a graduate student in my lab, alarmingly, who is from the computational sciences, Bart over there. He is working on machine-based learning approaches to improve how efficiently we can count things and measure things, and not just count and measure simple things, because we're very good at doing that, but relate multiple things together that happen in the same place at different points of disease progression. And it has a lot of potential to do a lot of things that we can't currently do. I think we don't fully understand what that potential will be, but we are very interested in using it to make our work much more efficient in multiple different ways.
So yeah, I've just written a paper on this, and there's so many different things. Craig's right. I mean, there's so many different things you can look. So what I will say, remember, there's a very famous phrase called garbage in, garbage out. AI is very much dependent on the data that you're actually utilizing. So when it comes to an individual called David Feigelbaum, who's like a champion in rare diseases, has a huge grant now on using AI for drug repurposing. That's hard science, that's chemistry, that's structural. AI can handle that. When it comes to the patient selection, their x-rays are fine. Now, you think every physician's notes that you use, they write the same things? Becomes very hard. So natural language processing is something that's very important there. So there's various areas and it will evolve at different speeds based on what we can actually do. Ineka, sorry,
I think you've opened Pandora's box with that question, because I don't think there's going to be any aspect of our life moving forward in general where AI hasn't touched it. And I think everybody here on the panel could tell you in their own little wheelhouse how AI is being used. And I've got another one. You just mentioned repurposing drugs. We're funding a study at the moment through the BDSRA Australia that is looking at computational modelling as well. And effectively what they're doing is taking the hundreds to thousands of FDA-approved drugs right now, looking at all the different proteins and genes and all of the different biochemical pathways that are affected or improved or touched in any way by those particular already approved drugs, and looking at the pathways through the different NCLs and seeing if there are any commonalities in those pathways, and looking through filtering processes can narrow that down very quickly without having to touch a pipette or a laboratory instrument, and narrow that down to a small number of hits, which they can then go and say, well, these are theoretically, these could potentially be affecting the same pathways in the different NCLs, and then they can go and take that very small number of compounds and go and test those in the cells and in dishes like you see up here, and then the animal models. So it's certainly used across the spectrum in so many ways. But watch this space, because I think it is going to explode. Like you say, it's a massive area, but there's also massive area for potential as well.
Yeah, I was just wondering, earlier was a discussion about how many compounds there are and how long it takes to get through IND. Is there a similar discussion or analysis done for how long it takes to get through drug repurposing trials?
That's a really good question, which I think there are many variables.
There's a couple of variables there, and I don't know if, Craig, you want to comment on this. One thing is drug repurposing is one thing. The drug often belongs to somebody, right? So first of all, hypothetically, you've got to get access to the drug and then the data. So to repurpose, because the most crucial piece is the safety data, right? And that's the really expensive bit early on before you get to the efficacy. So I don't know. What else do you want to add to that, Craig?
Well, I'm exactly right. I think if there is, and that's certainly been one of our targets, is trying to find companies who have molecules in very closely related conditions in neurodegeneration or lysosomal storage. They've gone through already having an open IND and or an already approved drug, so an NDA, filed and approved, if we can get access to that, they've already done all the IND enabling studies, which is a big part of that time frame and a big part of the cost. They've done phase 1, phase 2, probably phase 3. And so to add an indication substantially cuts time and cost out of that. And so now, our friends, and this is a whole new discussion if you want to get onto the regulatory agencies, because we touched on that yesterday with Dave. That's a whole new set of issues, but typically speaking, it cuts, years and anywhere from tens of millions to hundreds of millions of dollars off of the cost associated with, therapeutic development if you can find a repurposed drug.
One of the gorilla in the room is if a company has a drug and they have it approved for an indication and then you want to use it for another indication, they're very fearful that in a different population that there may be some other negative effects that would be associated to that would then ultimately that would maybe even affect their approval that they have. We ran into that with mycophenolate, with Cellcept, for example. They wouldn't give it to us. Okay, does anybody else want to add to that? Tom?
So we've got some work going on at the moment. We're looking at repurposing from something that's used for cancer therapy into motor neuron disease. And actually, because there's already 20 years worth of data on the use of these kind of compounds in human patients for that indication, as you say, what the tox and the tolerability is like, biodistribution. And so we just need to do 3 years worth of work to check it in disease-relevant models for motor neuron disease. And then we can put it into a multi-arm SMART trial for motor neuron disease. But freedom to operate, as you say, for a drug that's already available, if you've got freedom to operate, it's not a problem.
So taking the last two questions together, the way around this could be AI, because you're talking real chemistry, because you can use AI to slightly modify that approved drug that they won't give you. Maybe adding, just sticking a little hydrogen on there, or maybe something like that. Other questions? Big room to scan. I'm sorry I'm standing in the middle, but I have to, so if I'm blocking people's views. Other questions? Tracy, come on, you must have something. Yeah? No?
I can ask like a personalized infusion question. Well, my daughter has CLN2, and she has her biweekly infusions. But we started at the 2.0 rate, and then we went to 2.5 and slowed it down because she got sick. She's puking a lot. That helped with the vomiting, but not so much on her sickness. So like she feels like crap when she's done, and that leads to a seizure basically when we're walking out of the hospital. I didn't know if there would be anything else we can do. Right now we do Zyrtec and Tylenol before.
So if I think there's other people here also performing, I see the Colorado team. So what I do is we pre-medicate. So if Zofran doesn't work, scopolamine as like the anti-nausea medications that we will do, because if there's a patch, it's more long-lasting, right? So if you're in a cruise, right, you just don't take. So I think for what I say is you pre-medicate is key, and then you kind of chase. So that may be also a reaction to the medication, and that's when you were asking about immune suppression. So if you've tried all those pre-medication, Tylenol, Zofran, or Tylenol and scopolamine, or Tylenol and Phenergan, this also makes them sleepy, right? All this some medications. If you've tried some of that, that's when you bring, I call the big guns, so we can try steroids at that point to avoid the reaction, so they don't feel like, although there's some mommies here, I think a fair amount of the children feel like crap on the day off. And so sometimes when they have seizures, I allow that seizure for the day, or you can give Klonopin for that. So for example, it's that morning of the sedation, of the infusion, you give Klonopin, which is sort of our bridging dose to just relax them, because they get kind of like, you get tense, the kids get tense, and so we try those medications to kind of because it depends on what you feel. Like if you feel like you get nervous about the seizures, then you can give something like Klonopin or back or the medications like seizure meds.
Scott's going to add something here.
You actually covered most of it, so there's not a whole lot to add. I would just throw out there that we kind of start with a standard protocol for every patient with CLN2 when we start enzyme replacement, and every one of them we've had to then customize individually to them over time to make it as tolerable as possible and as manageable for them and the family as we can. There's no perfect answer there. We talk with each other. We say, oh, well, have you dealt with this? We'd be happy to connect with your physicians if it's helpful for them to connect with any of us and chat about creative ideas on how to make things better.
And you also get 2 bites of the cherry because tomorrow we're going to have an Ask a Doc session too. By all means, ask questions now too, but we have Centres of Excellence directors floating around conference this weekend who are all experts like Dr. Demarest and Dr. Delos Reyes. We have a panel of 6, so please bring those questions there too.
And any more questions from the audience? Okay, there's the Ask the Doctor. Oh, Tracy didn't let me down. We got to go at least 1 minute over because otherwise Amy won't give us this much time next year.
So this is just an inquiry question, maybe directed more towards John. I noticed during the animal models portion, there was a CLN2 monkey colony that was talked about. I think that's relatively unknown and new. What is the realistic utility of that colony, and what is the communication with the group that has that colony currently?
Okay,
Great
so
question.
we are already in contact with the people in Japan. We have set up a collaboration. We've exchanged all the bits of paper we need to so that we can move forwards with that collaboration and get some data from that model, which is yet to be properly characterized to know what it is like. We would then use that information to as preliminary data for getting larger funding for doing bigger studies within that. So they have identified this model. They have very limited numbers of them. It's a question of then knowing what we can do to expand that model and make it actually a model as opposed to some isolated cases that have been identified. But the first stage will be that we need to actually characterize it and know what it actually looks like. That is underway, and that will go ahead with, and we're talking to them with a view of doing funding applications to make it a useful model.
Yeah, I was just going to add to the kind of the commentary generally about the repurposing of drugs and looking at things that are in nearby diseases that are really closely linked to Batten. So what we found, I think, either for something that's already approved in the one case, a drug, and then in a second that should be approved soon, I think, for another related condition to Batten, we've actually found those companies, because their drugs have a really good, to Dave's point, they're not as worried about the safety and tolerability of their compound. They're very open to adding an indication to their pipeline because it's quicker, easier, and it's almost like found money for them as a pharmaceutical company. It really wins for everybody. So what that then enables is, like ourselves, we have 2 current projects going on where they're donating the drug. We're testing it in, Batten models to see if it translates from that other lysosomal storage disease or that other neurodegenerative disease, and we can see the same benefits in Batten models, and so that would be a very easy addition for those pharmaceutical companies to then, add to their programs. And so I think on the hopeful side, there are either things that are already approved in these closely related diseases, or we think is coming pretty quickly that I think can reasonably, with some speed, be added to the Batten disease portfolio.
Okay, I think we are going to close out right now.
They're adults.
No, I said it was you.
Thank you all very much for coming. Lunch is served in the hall next door, and we're all here floating around for a couple of days, so please come talk to us anytime. Thank you.